Welcome to episode 195 of the Reptile Gumbo podcast. It's a very special episode. It's a Christmas miracle. We'll get to it in a second. Let's do our
Christmas miracle because I'm upright.
that is true.
This is why babies cry with ear infections.
So you're saying you're a baby?
I have wanted to die since Friday.
Yeah, I agree. You're a big old baby.
It's awful.
I get tired of people saying that guys always like get upset when they get hurt or they're sick. I don't ever cry.
You're really not. You kind of like completely shut down when you're sick.
You
So,
also
like,
don't
you
realize
sleep a
that
lot.
I'm constantly hurting or
But-
ill of something.
I think the hardest
life.
part for me the last 4 days has literally been just being down and not doing anything.
Yeah, no one's made my dinner,
You
washed my
have
dishes.
done it all.
Shit sucks.
It's a Christmas miracle.
Married you for a reason.
I
need you to
know.
get healthy again.
I'm slacking off. while I'm
It's gonna go way back to that guy like 100 episodes ago told me I need to be nicer to my wife.
Ugh.
Fuck off, that guy. Anyways, it's the two of us tonight. Robert is, speaking of being down sick, Robert is down.
Yeah.
Hopefully he will get better, but he is resting because he needs to rest. So it is the two of us.
Yeah.
Want to do our sponsor stuff and then get on to the show? Maybe?
Yes.
Possibly?
I had the wrong notes pulled up on my phone because somebody's kid sent me their Christmas list.
Somebody, it's your kid.
Your kid. She's not mine today.
Go ahead.
If you're looking for a high quality PVC rack, look no further than Lone Star Reptile Racks. They offer a variety of sizes for all types of snakes, geckos, rats, and more. You can even order something custom. Shipping is available, or you can plan to pick up at a Herbst Reptile show near you. Visit ellisreptileracks.com to reach out to Lone Star Reptile Racks and place your order today.
Yes. And Little Shop of Horse. Little Shop of Horse is a small feeder and pet supply business based in San Antonio, and they regularly schedule feeder meetups around San Antonio as well as other neighboring towns and cities. They offer shipping on their feeder insects, ice pods, and are working on starting shipping on their feeder rodents too. All feeders are raised on a nutritional diet that optimizes the health of the reptiles and the amphibians that consume them. Give a shout out to our friend Lewis over at Little Shop of Horrors. Get you some feeder insects. If you're in the area, some feeder rodents. They're great. Talk to them. I pull up. I got my Herbst Reptile Show dates pulled up here. So let me go ahead and look. I even prepared. Look, I got little tickers in across the bottom of the screen. Bam.
Look at you.
I know.
So fancy.
Technology I've always had, never used. Herbst Reptile Shows. Oklahoma City, Oklahoma, January 13th and 14th. Not going to that because Oklahoma is a different planet and it's too far away from here. One of our guests no longer lives there. So Longview, Texas, January 20th and 21st. Then the Big Conroe Show, January 27th, 28th, which is my birthday weekend. Come by, see me, say hi. It may end up just being me that weekend.
No. I should be there for some of it.
Okay. Come by and see us. Corpus Christi, Texas, February 24th and 25th. Over to the big show in Baton Rouge. I say Baton Rouge. It's not Baton Rouge. It's Gonzales.
Mm-hmm.
It's Gonzales. Anyways, Baton Rouge. March 2nd, March 3rd. Then Bryan College Station, March 9th, March 10th. Then Rosenberg, Texas, which I was confused on where that was, but that's like 20 minutes down the road. I didn't realize that because
I've never heard of it.
it's the other side of Sugarland. So if anybody knows where Sugarland, Texas is,
Oh,
that's
okay.
Rosenberg, Texas is March 16th, March 17th. That's a home show for us. Slidell, Louisiana, April 6th and April 7th. So make it to those shows. They're all next year. So if you haven't gotten your Christmas presents yet, you should have a look. What?
Joe's going to hand me the paper towels behind you, unless you can reach them.
You mean the ones right here?
Yep. Can you? God, your arms are long.
They're like 2 feet away.
Leave me
Did
alone.
you text your kid from the other room to come get something that was 2 feet away?
I did because I didn't want to interrupt
Good.
you.
She had to get up. That's good. You know, she slept till 3 today. So make her do something.
It was 4:30.
Well, kids suck. Also, VivTech. If you haven't gotten your LED UVB light bulbs from VivTech yet, what are you waiting for? Go ahead and get those and use code GUMBO22 to save 15% on your VivTech products for either your light bulbs or smart products or anything else that VivTech offers at this point. Go there, get some bulbs, buy some bulbs.
Okay, so one of our guests was not kidding. This headphone on my ear is fucking murder.
Well, yeah, it's
I don't know why I thought it wasn't going to be a big deal. It's pretty brutal.
well, leave that off your ear.
Yeah, I am.
Dingbat.
I have a severe ear infection.
For anybody listening, we do have our giveaway for this week. Well, technically, it's last week of December. The last week we did. We're a week off. But we're giving away something every week of December, thanks to our friends over at Colossal Constrictors. The first week we gave away the Red Science Scraper Set. This week we're giving away the Red Science Probe Kit.
Oh, that's cool.
So if you're listening to this live right now, go over to our Facebook page. Tell us what you would like for Christmas. It's on the pinned
Come on, there's
post at the top.
14 of y'all. I think that's like our highest number ever.
It's not the highest number ever. It's overreacting. But go tell us what you want for Christmas on our post that's pinned at the top of our Facebook page, and I will put you into the giveaway at the end of the episode. There's also a giveaway next week for some reptile bedding, and then I think the week after that is for a digital scale for those of you weirdos that weigh out your snakes. I'm talking to you ball python people. No one else weighs their snakes. We don't know what they weigh. Don't ask anybody else that breeds any other snake. We have no idea what it weighs. We also don't know what their food weighs. We don't know how much their shit weighs. None of that stuff. That's just you guys. All right, let's go ahead and bring in guest.
Go ahead, man.
All right, so we got a lot tonight. So
Y'all just don't even know.
I'm gonna bring them in, and then I'm gonna let them introduce themselves out past their name because I'm there's too many letters and stuff past their name. I'm not gonna be able to get all that.
Christmas miracle.
We're gonna make this one wait last.
No.
We're making them wait last.
Nice.
Let's go in.
He knows it's him.
with Dr. Zac Loughman. Let me get all of y'all in, and then y'all can introduce the letters after your name. And then Dr. Warren Booth, Dr. Ben Morrill, and Dr. Travis Wyman. He's here.
Oh my goodness.
How's it going, Travis?
I don't think James has been this excited about a podcast that he's had scheduled ever since like the very beginning when he started getting people.
I didn't know if it was going to happen for one. It happens.
Y'all made his Christmas. It's hilarious.
All right, cool. So let's let everybody introduce themselves. Everyone here has been on the show at some point, but we get new people all the time, and some people don't listen to the other 5,000 podcasts they've been on. Some of them have their own podcast. So let's go through everybody, and then we'll get into some discussion. So, Zach, you go first.
Oh, okay. I'm Dr. Zac Loughman. I am at West Liberty University. It's a little school in northern West Virginia. I've been on a bunch of podcasts. I have a podcast, Colubrid and Colubrid Radio. And I have grad students do herpetoculture stuff, herpetology, but then the crayfish, it's over here, I guess. That's what I'm known for professionally, I guess. So there you go.
All right. Warren, you're next.
Warren Booth, Dr. Warren Booth, if you want that, at Virginia Tech. That's a really big school in west of Virginia, not Western Virginia. Evolutionary geneticist, evolutionary genomics of how organisms adapt and evolve in urban environments, but I also do a little bit of work on parthenogenesis and sperm storage in reptiles.
All right. Dr. Ben. Your volume's not on, so it'd be kind of hard to hear. Nope, still not. You.
Sad day.
He puts out videos every week. You would think you would have
That's
this.
The problem was you're like, you should use headphones. It'll help with sound. I've never tried it before. Apparently, it doesn't work, or I don't have it set. So no headphones for me.
Right.
Yeah, Dr. Ben Morrill, Rare Genetics Inc. Working to design genetic tests for reptiles. Trying to get as many tests for as many species and bring costs down as much as I can when I can, and I'm just excited to see how that can help us as keepers and breeders, and maybe help us learn a little bit more about the animals that we keep.
Awesome. And then Travis Wyman.
Don't be ugly. Quit being ugly.
This is just the way he is, Katie. You know that.
Man.
I know that.
He made you the savior in that picture.
Yeah, you got to be baby Jesus. Come on.
Good point. Make the atheist baby Jesus. That works great.
Another atheist made the whole meme. So.
It's okay. He would show me stuff and I'd be like, too far. He's like, it's not too far. I'm posting it. I'm like,
Travis Wyman. I work as a government contractor for Battelle, through Battelle, I guess. I am a forensic genomicist, I guess would be the best way to describe me.
Most confusing way to describe it? Got it. Okay.
and I teach 4th grade.
and I try to teach kids.
I'm going to be Googling so much shit tonight so that I understand what the hell we're talking about. It's going to be great. Between the medications and between tonight.
I don't know about Ben, but at least 3 of us have some teaching, tutoring experience. So we know how to not just act like it's a...
You can dumb it down for me,
level.
Travis. I won't hate you for saying that.
It's gonna work. I got to teach genetics when I get back after Christmas break. We do genetics, so I get to teach kids how to do a Punnett square and fuck that up over and over again.
And I got to teach intro bio and anat and phys to community college students, and then be a TA as a grad student, of course.
So see, there you go. All four of us have some degree of teaching experience, so we can make it on a level for everybody.
Ben says he got to teach like it was an honor. Come on, then you haven't taught long enough. It is not an honor.
Hey, I like my job.
Because you don't teach high school senior or freshmen.
Nope, I teach babies.
No one here except for me suffers through high school freshmen.
I love my babies.
No, I don't like any of mine. I told them that. I feel like you gotta be truthful with them and tell them.
The honesty is key.
They have it in their head that teachers are supposed to like them. I was like, I don't have to like you to teach you. And they keep asking me, why am I here? I'm like, they pay me.
I do tell my 10-year-old sometimes that I may love them, but I don't like them at that moment. And then they need to fix it. And then I tell them to walk away. And then normally they get it together and then we're fine.
Still waiting for a freshman to get it together.
Yeah, good luck with that.
Yeah, it doesn't happen.
Yeah.
So I do want to ask, so talking about teaching, I do want to ask, since several of y'all, we've all taught, and y'all are teaching at college level, how many of y'all deal with college freshmen?
It's been a long time.
As a grad student,
I deal with all of them. Freshman through second year master's student. I don't have PhD students here. So
are you finding
I don't teach at the moment. So.
Way to go.
I won't be teaching until 2025. So I went from teaching 6 courses a year to teaching 0 courses a year, which is awesome.
I would say this, and you're not complaining.
Zach is jealous.
Twisting the knife.
So,
but I'm happy for you, Warren. You know that.
Look at that. Of course you are.
with freshmen coming in, and you've taught long enough, have you noticed, are they less and less prepared for college at this point?
Yes.
Okay.
Unequivocally. I have only been teaching since 2006. And it's funny because when I came in when it was like millennials are the end of the world. The world is going to come to an end. And now I'm teaching Gen Z. And I'm not going to bash on Gen Z and I'm not going to bash on millennials. I'll just simply say it's a very different mindset of the role of the teacher and what the teacher's supposed to be in your life. And, before when I started teaching, somebody would fail a test. The majority, how do I say this? Because the majority didn't fail the test, but the people that were getting like C's, D's, F's in biology, they would come to you and they'd be like, okay, what do I need to do better? Now they come to you and it's kind of this, you're ruining my life. How do the onus is on me to make their neurons work, which I don't understand how to do that. But I don't want to paint the picture that that's all of them. That's like 20% or less, because I also have plenty of freshmen, sophomores, juniors, seniors that are like kicking ass and taking names. So it's just the response to failure is very different than it was back in 06. That's the thing that I noticed that's different.
I guess also the benefit for teaching college is like they're paying to be there and they had to have grades to get in there at least. Whereas like teaching in high school, like the government's telling them they have to be there and they're forced to be there. And I've taught since 2013, and I've noticed just in that 10-year span, I've noticed a huge change in education. And I just wonder how unprepared these kids are at the next level.
It's funny. The neat thing about West Liberty is, historically speaking, we just simply receive students from Eastern Ohio, Western Pennsylvania, Northern West Virginia. I mean, we're like a little liberal arts school. So those schools, normally the feeders populations are the local schools. But with our ZooSci major, we're one of 6. Like, that's it. And so we've suddenly gotten all these people from across the country, which is funny. It's a little bit of culture shock to West Liberty, to be honest with you. Like, what do we do with these people from California? They think very differently than the people from Wheeling.
Wow. I bet it's a culture shock for them too.
It is too, because our university is literally in a cow field. Like, if you look at it on Google Earth, we have had cows on campus.
Nice. That
have broken away. They're literally less than a mile away. But it's been really cool for me as an educator, because I get to, I'm receiving these students from all over the country now, and seeing how the different regions respond to like me as an instructor has been really interesting. But I don't think you can paint with a broad brush, but I do think there are at the same time general trends that are somewhat different. And COVID COVID didn't do anybody any favors because there was a bit of a role flip. And you guys know this, you teach high school. My wife's a high school teacher. We were told to cater to the students, obviously, because of the COVID landscape. But like what little, I don't want to say power, but that's probably the right word, that educators had, it kind of just went out the window because, you know, you're teaching through a computer and you got to do projects and people got to turn things in and you got to be empathetic to everyone. And then, but the students figured out very early on, like, oh, we can milk this. So it's just been a really weird world since 2020. We'll just leave it at that.
Yeah, the group of fourth graders that I have now is the same group of third graders that I had last year. I looped with them and they didn't have kindergarten or first grade. So there were no social skills. So they were completely feral when they came in as second graders, if they came to school and weren't homeschooled. So a lot of them, their first time being in school was when they had me last year in third grade. And I don't teach kindergarten for a reason. I don't know how to teach those social skills. They're looking at you, turn and look the other way. I don't know what to tell you. Like, no, it was hard. It was very hard.
It's funny. That actually reminds me of something. One big difference is this wonderful device here has eliminated everybody's ability to communicate with each other. And like conflict resolution, things that I don't even think are conflicts. Like, we've got to figure out how we're all going to get in the van. That leads to drama. And I'm like, what the hell? Just get in the damn van. Like, but no. Trust me. It dilutes a little bit at my level. Now, granted, I'm at a liberal arts school. I don't know if it's different at a bigger school. Warren was at a bigger school. So he might be listening to me thinking he's full of crap.
No, Warren said, fuck it. I'm not teaching now for the next couple of years.
Warren is enjoying no students.
Warren said, I have freedom now.
The promised land. But that's the thing. And then I became a chair, which means that everybody's problem's my problem. So I get to hear like, this person's being mean to me, this person's being mean to me, blah, blah, blah. So it's just been very, conflict resolution is not a thing anymore because we just get on the phone and
I'm trying to teach it in fourth grade, but it's not working out.
We're doing the Lord's work, so keep it up.
What kills me about having a phone in their pocket is like, I'll have my kids working on something on their computer and they'll look at me and go, how do you spell this? I'm like, you have the internet in front of just think for a second.
Type it into Google. It'll tell you if you're wrong.
I'm like, they're like, well, if I don't know how to spell it, how do I figure it out? Try your best. Google will fix that for you. Come on. It's problem solving skills is what I have found.
Working on that too, guys. Working on that too.
Yeah. That's my 2 cents on it.
So Warren, what are you doing now that you don't have to be around peons?
Just all the research. So I've got, I will teach. I've got a graduate seminar. So my department that I'm in is just a graduate department. I will teach, I think January 2025, I've got to oversee a graduate seminar. And then teaching is maybe one class every year or 2 years, depending. But other than that, it's research. So I've got a lab with grad students, postdocs, and that's about it, really. You know, so I don't have any
than lectures.
Yeah, I taught 6 courses a year, and that was just overwhelming. And I still had state and federal funded research, and I still had postdocs and grad students. So it's nice taking away that whole aspect because, you know, at this point in time, normally I would be grading and drinking really heavily because of the grading. Now I'm just drinking.
But now you're just drinking heavily,
which is great.
I've never met a career that drinks as much as teachers.
Well, that's because students drive us to it.
They do.
11 years at it at my former institution, and now it's just, you know, it's the downhill slope, you know. So
I mean, I had a whole bottle of wine on Friday, so yeah.
I don't drink, but I just cuss a lot. That makes me feel better. And then I try not to do it around kids, but that doesn't happen either. I cuss at them also. What I find interesting about the 4 of y'all, and now that Warren moved, is that all of y'all are like within a few hours of each other, basically now at this point.
Yeah.
Which is the weirdest thing that everybody's like, Virginia, West Virginia. That's where we're all going to move. All the doctors are there.
In fact, I had lunch with Ben like a month ago.
Yeah.
I saw Zac, like a month ago?
At Tinley.
Never met Travis.
Fuck off, Travis. He don't want to talk to you.
I've driven around that area a lot in the last Like I was in DC last night picking up my in-laws. I was in Virginia Beach in Norfolk 2 weeks ago. 3 weeks ago, I was in DC doing something else. So I'm in that region a lot. So eventually, we will get to meet up. Probably
eventually we're going to meet up.
Fest
I
or
mean,
something.
it's an inevitability.
So all I hear right now is
Well,
Warren's
probably
been dodging you.
Well, that's pretty much it. No, I can't avoid that anymore. So
I'm trying really hard to convince our external speaker committee that we want to have Warren come up and talk about stuff, because I think that his urban pest stuff would be very interesting just as a field for my people to listen to.
Thought you're just gonna have him come talk about like boas and stuff. I'm like, I don't think it's gonna work that way. That's your job. Just
Some of them might be interested in the parthenogenesis stuff, but I think a lot of them would be very interested in that urban pest ecology. That
bed
Warren
bugs.
looks at.
That's all I can think when Warren is bed bugs. I've never had bed
bugs and
bugs.
cockroaches.
Fun fact
About bed bugs.
about bed bugs.
You currently
There-
have bed bugs.
No. Don't put that out there in the universe. I'm dealing with enough right now with this ear
That's
infection.
not a fun fact.
No,
Or a horrible fact.
there's bloodletting.
No. There is a environmentally friendly way to kill them
Bomb.
with heat.
A bomb. Nuclear bomb.
And you have to vacuum seal all the windows, and then you have to pump heat. No? Why are you shaking your head?
Because
No.
he knows about bed
Are
bugs.
you telling me that
Yeah,
I suffered for hours of going in and checking and making sure that cabin didn't catch on fire, and it was for naught?
Yeah.
It was the longest day of summer.
So she's a summer.
No,
She
they
works
do
at a
that,
camper
though.
in the
Yeah,
summer.
it's like if it's in a single family home, they'll do it room by room, and they'll normally heat it up to about 141 degrees, but they'll use special heaters that won't catch on fire, and you don't need to seal anything. The problem is that if the building's too large, what you get is a heat gradient. So away from the heat spot, it then gets cooler and cooler and cooler. So if you've got any in wall voids or on the floor or in beds that are away from that heat spot, you'll actually cause them to disperse further out. So you can actually cause them to spread further within your house if it's not done properly. And if you try to do it on your own if you try to do it on your own, you go to like one of the big box stores and get a propane heater, you're more likely to burn your house down.
Oh yeah,
That's
no,
also a
this
very
was a
common
company. This company brought this thing into the summer camp. The campers were convinced that there was an ant infestation, and
That's what you lied to them.
the
and told
kids
them.
were all moved to a different location. And I don't know how I drew the short straw, but I hung out on that front porch all day. And every 20 minutes, according to the alarm, I had to go in and make sure that all the probes were still hanging in the right spot upstairs and downstairs. And the thermometer got up to 175 in that cabin.
Was the company not there to do it for you?
Apparently, it was going to cost a whole lot more money for them to have someone stay on
To do
site
it
all
properly?
day. And the guy was
We're
like,
just gonna send little
all you
kids
gotta
in
do
there
is
and check the
check it.
temperature.
all you got to do is check it. Katie's smart. She can do it.
That's
Let Katie walk into the building and just die.
It was awful.
Enjoy your bed bugs because they ain't going away.
I never got them. I didn't live in that
I also
cabin
love that
this
you just
summer.
tell the kids it was ants instead of
Yeah.
one of you little fuckers is nasty and brought bed bugs.
It was rough.
The ants are
It was
really easy
rough.
to control. Bed bugs are a lot harder.
Yeah. That's my only experience with them.
It's funny though because there's a parallel like in the hobby because I think bed bugs and then I think like when we're snakes I think mites. Like I feel like it's the same issue. Like both are hard to
Mites
get
are
rid of.
good
That L word
Yeah,
we're not allowed to use in this house.
Mites are going to
mites.
become a bigger problem. We're seeing a lot of resistance evolving to the common insecticides people are using. So people normally use pyrethroids
Yeah.
or permethrin based, and they evolve resistance really, really quickly. So I've had samples sent to me, and we've identified mutations in sodium channel genes that are associated with resistance in snake mite
that's
populations.
Oh,
And given how
Wow.
stupid people are whenever they get new snakes and they don't quarantine, no matter how much you tell them you should quarantine, it's going to become a major problem.
So what I
Heard it
hear
here
is
first.
I'm never getting my closet back.
No, there's snakes go in there. It's just after about 3 or 4 months, then they can finally move out.
The
I'm
bed
never
bugs
doing my-
and the mites will fight it out, so they will. It'll be great. They'll just
What
form this
about
harmonious little community?
what about the active ingredient in Frontline?
Which one is that?
I don't
remember Nick's.
I know it's different. I'm pretty sure it's not.
Nix is the
Nix
one
is
for
permitted.
head
No,
lice.
Nix is
it's one of the nicotinamide analogs.
We don't
neonicotinoid.
use the L word in this house. It makes my head itch.
A neonic. So we,
It's like
we
my
see
biggest
resistance.
fear as a
We see
teacher.
resistance to neonics as well, but not. I haven't seen it in snake mites, but we see it in bed bugs, cockroaches, and stuff like that. There, so they will if people start using that a lot, we will see resistance evolve in time, and where we will see it are in really big, large breeder facilities that have got or large importers that have got a lot of mites. Because if they never control them, then you know what it's like. Well, all of you, the more individuals there are reproducing, the more chance there is for a mutation to arise within that population.
Mm-hmm.
And whenever you're hitting them all with the same insecticide, because of the large population size and likelihood of mutations or mutation rate, you're likely to find individuals resistant, and they will then thrive and become the dominant genotype in the next generations coming. So we will see neonic resistance probably in the next handful of years as well.
I actually have a question about that. How does so you spray that on the snake or you wipe it on the snake? What's the mode of action that actually kills the mite? Like, does it basically create a film on the snake that the chelicerae go down through and then that's it? Or is it in the bloodstream? Like, I've never really taken the time to learn how that actually works. And I'm sure if...
So when it's on the snake, then it should have a residual effect. And it's just, it'll be soaked through the cuticular, the cuticular kind of segments. And that will absorb into the hemolymph that will then spread around. And it'll have the same mode. I think it acts on a nerve channel as well.
Cool.
I just saw Alicia ask about fipronil. Fipronil, there are resistance. There's a mechanisms. The GABA receptor gene has a resistance to dieldrin mutation. The dieldrin and fipronil have the same mode of action, and we see fipronil resistance in German cockroach populations. So again, it's because it can be a target site mutation. It's likely to be one that we see pop up in time, and it takes time to spread. Any of the target site mutations, the pyrethroids, permethrin, that kind of thing, fipronil, dieldrin, that kind of thing, you'll see target site mutations arising.
And so I guess the challenge is going to be
but it
finding
is fun.
something.
Kind of cool. The research is fun. Yeah.
The challenge will be finding something strong enough in the future that takes care of them, that doesn't also kill your animal.
No. It's going to be that meme with the little girl that like burns her house down.
Just light everything on fire.
Yeah. Burn it
Unfortunately, with heat, your snakes won't react well to that either.
Yeah.
So you will kill the mites, but you won't have any snakes either. But you will have a lot of jerky, snake jerky with dead mites. So
mites peppered in on it.
I think one of the areas to look at are the predatory mites.
Yeah.
I've never had to use them. I haven't had mites in my collection in over a dozen years, but I think the predatory mites are the ones that you want to look at. I've heard really good things about those.
I inoculate. I use naturalistic setups, and I inoculate them just preemptively with the predatory mites, not so much as a mite balancer, but because when you first set up any kind of naturalistic setup, kind of the first thing you get is that flash of fungus flies and stuff, and the predatory mites will help keep those down.
Yeah.
So wait, so you're actually introducing a mite?
Yeah, but they're not snake mites.
To prevent
these are mites that eat mites.
They're mites that eat other invertebrates.
Gotcha.
So
that's cool.
I don't know. It makes my skin itch when I just think about putting other mites into a cage. And I know in my head they're not snake mites, but I'm like, after when we moved to Texas, I had mites for the first time in a decade and a half, and getting rid of those were a nightmare. And so just the idea of any sort of mite again that I'm putting into my collection on purpose scares the ever-loving crap out of me.
Well, they look totally different than a snake mite. So you at least...
Do they? Okay.
Yeah, they're like reddish-orange, one of the species.
All right, time to start.
The only issue I've had in getting them
striped,
is that they mine have always been dead on arrival. Like I haven't been able to find a supplier because they're a little bit temperamental. But that's also just me being lazy and going to the first one I found.
Oh yeah.
I mean, they're cool looking.
Yeah, they don't look anything like what the others did though.
It's might scale. I know. Jason asked, how long does frontline or any of that stuff stay on an animal? Not counting a shed, how long does that stuff stay on there? Is it become part of their blood for a while, or what is it?
I don't know. So what with most animals, like if you use it on dogs, it's normally about a month, right?
Yeah.
So I would imagine it's going to be several weeks. But how temperature and humidity affect that, I'm not sure. As to say, I haven't had to use it, and I don't plan to ever have to use it, and I don't. There's no reason to study that in the lab. There's no. I don't think National Science Foundation or USDA are going to give me much money to study snake mites.
Yeah, probably
To
not.
do research in my lab, it costs a lot of money. Yeah.
So Zach, how much of that is how often are y'all bringing new animals into your collection with your zoo sciences and all?
Not that much now. We were really heavily doing that in 16, 17, 18, and we definitely had mite outbreaks. My problem is I think that I have students that have mites that then get them on their little fingers and then come in and work with our collection, because we get these we'll get a random pop-up.
Yeah.
And it doesn't make any damn sense. Like, we're talking about animals that have been living in a rack or a stack of PVCs for like 8 years, 5 years, nothing. And then here they show up. So we're hyper vigilant, because that's the really interesting thing about this collection at the school is it's kind of a nightmare for quarantining, because like we do quarantine, but then I've got
I love the air quotes.
Well, I say that because we do it, but then we have 12 people working with the animals. And then of those 12 people, 4 of them have snakes. So, you know what I mean? And then when it gets put into the collection for teaching purposes and training, we have just over 100 undergrads. And of those, I would say at least 20 of them are like beyond, I have a bearded dragon, like kind of like us. And so, and they're 19, 20, they try. Many of them are excellent. Some of them are just starting, and they go to the local Pittsburgh show. And I mean, I would go to that show to get mites. You know what I mean? It's one of those at times. I just go there to get feeders when I'm desperate. But when I hear, oh, I was at the show, and then I'm like, oh, it's Tuesday, and you've been in the collection. Here come whatever the hell. So, but what it has done, it has kept me on my toes. I have seen a lot of things go down with this collection that I never thought that I would have to interact with.
So my first experience with mites was in college. I had a snake before college. I brought it to college. It did not have mites before that. And then I kept it illegally in my dorm room, as most people do. And then it just kept getting mites. And there were no other reptiles in the room other than my snake. And all I can blame is my 60-year-old dorm, which was nasty as hell. Like, not my room, just the actual building itself. That means there's just mites living in this dorm. Snake mites just crawling through this dorm at some point, apparently. And I could not get rid of them. Finally, I had to be like, all right, all my snakes have to go back to my parents' house. My parents had to take care of them until I moved out of the dorms again.
He also could not do his genetics project in his dorm room because they kept molding and dying.
Yeah, every time I opened a fruit fly culture in my dorm room, straight mold would grow. But if I opened them in her
do
dorm
that in your dorm room?
Yeah, on the weekends, like to try and
they don't react. Yeah, they don't react well to weed, and they don't react well to general student dorm conditions.
My dorm was fine.
Yeah, I'd have to go to Katie's dorm to do all my genetic stuff with my fruit flies because the air there wouldn't cause mold to grow. But I was like, God, how bad is the air in our dorm building that every time I open this, I kill fruit flies? And then I just finally at the end of it, I was so tired of it. I said, fuck it. I found a friend who had basically the same genetics for his fruit flies. Like, hey, give me your numbers. I switched them a little bit. And I was like, all right, let's turn that in. That works.
Wow.
That's why I can't
Such
do
a horrible
That's the kind of
That's
student
why
you deal with.
See,
That's it right there.
hey, but I was smart
Right
enough
there.
to problem solve.
Like,
I problem solve.
all your tables are off by 2.
It still worked.
Gee.
This is why I would love to have something like dart frogs, but that whole thing just scared me of fruit flies, and I can never do it again. Fucking genetics.
Can't trust
That
a
was a
word
rough
this guy
class.
says anymore. It's fed 4 times. Oh, has it? It's fed once. Yeah.
It was horrible. Fruit flies and mites, 2 small things that annoy the crap out of me. I keep getting questions in for.
I know. I'm fixing to pull it up.
I've got them all pulled up. We can go through several of the things that people want to talk about. One was our buddy Paul Byfield said it was genetics related. He wants to know why his ex-wife was so angry all the time. Sorry, Paul, we can't answer that one. That's all on you, man.
It's all hormonal.
Some of these were asked, and I was like, "Ooh, I know the answer to these," but then I was like, if I answer them here, I'm going to be told while I'm wrong, and then corrected like my students who think they know the answer to shit.
Come on, man. Let's do it.
So I'll go with this first one. These are basically all genetics related because y'all are all pretty much genetics related. This is our friend Tracy said this might be a dumb question, but she's been learning a lot about quail genetics. We'll get to reptiles, but with them, they use the location of the gene along with what it does. So because of the location, one bird can't have multiple genes that are located in that one spot. It made me realize I've never heard anyone talk about the gene location with reptiles. Is this something that doesn't matter with them, or is it just something that really isn't studied? Which I'm going to let them answer it, but she just doesn't realize she has heard this because.
Have you told her this before?
No, but she's been around people that have ball pythons, and like it's an allelic thing. Anyways, so I figured I would let y'all.
Not me. I am the only ecologist.
Yes.
The only reason it's got an issue is, the only reason it matters is if it's sex linked, so it's on the X or the Y chromosome, or if there are two genes that are really close together, and there's a likelihood of those two genes being inherited in the same block, right? So say you had striped and albino that were right beside each other, the likelihood is that during crossing over they could get inherited preferentially together. So it'd be very difficult to get an individual who was one or the other. Other than that, it's not a huge deal which chromosome it's on, other than a sex-linked chromosome. Like, what's that one, banana or coral glow, or
Banana, yeah.
I don't know about other sex-linked genes. We spend a lot of time annotating genomes. So when we sequence genomes to chromosome scale, we spend a lot of time working out where the genes are, but we use that for totally different things in kind of urban evolution. But for
And
snake
I think
genetics,
that's
I don't know
kind of along the
what
lines
Ben
of
thinks
what
at the
she
moment,
was
but
asking.
Yeah. So
Is
for
why
the
don't we
only
do
thing
that
that's
with
important for me is
reptiles?
hitchhiking selection. Yeah. Because there's no real need. I don't see it anyway, unless there's these two genes or three genes that are in this block that are likely to be inherited together, then at that point it would be hard to disentangle. It would be hard to separate those genes out. So I don't know. Ben might be able to comment more on that as well.
If what she's talking about, like you were saying, James, if it's something to do with being allelic, then yeah, you can have different alleles for the same gene that maybe have different phenotypes. It looks different, kind of like the yellow belly complex in ball pythons,
which would be located in the same
blue-eyed
place.
Lucy.
So
there are
different variants of the
mutations
same gene.
mutations in the same gene. They're just in different spots of the same gene. So they're not identical, but they are mutations in the same gene, and that definitely can happen in any animals or plants.
So I think what people need to realize is that with the mutation in a gene, the gene's coding for a protein, right?
Yeah.
A mutation in that gene can either have no effect, right? So it doesn't do anything. Most of them will be that. Or it can have an effect and it can change an amino acid, and that amino acid can then change the shape that protein forms, or the variant that produces. You can have mutations. Let's think of, I don't know, some of these ball python ones, right? Where if you cross them together, you get a similar variant. Different mutations can have different phenotypes because they either completely stop the production of the protein, or they change the protein that was produced, and therefore that shape. So it might have a slight effect, but not a complete effect. So therefore, when people talk about things being allelic, just remember that they're not stopping the gene working; they're changing the variant. And in some cases, it could stop the gene working, but in other ones, it can just change that variant slightly, where it binds to a lesser degree later on down the chain and has a limited effect or a change in effect. But whenever you have two mutated versions coming together, they create a mutant phenotype down the line.
Well, and like with this, like I said, I think Tracy's heard this stuff before, but because the different hobbies with birds, reptiles, fish, and all the different things, and then most folks that are breeding and keeping these hobbies are not people that study genetics, and so now they're trying to talk about genetics from a purely standpoint where they've never actually understood genetics prior to this. Bred to this makes a white one, and like that's what they got. It gets very confusing, even though we're talking about the same thing.
Yeah, the other thing is that in things like quail genetics, just like in cattle, there's a lot of work put in to maximize production, and therefore they want to know the genome of the individuals. They want to know what's happening whenever different genes are in different forms, because they want to select for the individuals with the genomic combinations that result in the utmost production or the highest quality, meat or individual, and so on. So it's very different than what snake breeders do. Down the line, of course, it could come to that where people think that variant X is better to have in this thing and variant Y is better to have in that. But at the moment, it's not a huge deal. But it's very different in the meat production or agricultural kind of setting, and it's used for a different reason.
And hand in hand with that is it's been going on for so long with them that they've got a much bigger baseline because people cared, so they did a lot of sequencing to understand it. It's only the last couple of years with Ben that people have actually started to care about these things in snakes and stuff. So
look, we're still struggling to get people to understand the difference between incomplete and codominant. So
and
You always will.
We also have the awesome terminology of a super this and that.
Yeah.
That certainly didn't come from a geneticist.
So-
and the hidden genes.
Yeah, hidden gene. That's even worse.
So the next question, I could answer this one, but I'm gonna let it go to y'all. It kind of ties into that because we don't use. I find it funny.
Is this the 66%
Yes.
het question?
I find a lot of people use terminology in snakes that they don't fully understand what the term means. And this one is about het. And I think 90% of people that use the word het don't actually even know the long verb, like what it's short for. But the question was, where does this 66% het come from, and why do many prominent breeders use the term? Who wants to explain percentage of hets to people?
So study your Punnett square. You got a single gene, just put it in your Punnett square. 1 in 4 will be homozygous recessive. 1 in 4 will be homozygous mutant. 1 or 2 in 4 will be heterozygous. The problem is that for the 3, the one that's homozygous recessive and the 2 heterozygous, you don't know which ones of those 3, if it's non-visual, which of those 3 are actually heterozygous and which aren't. So therefore, 3 out of 4 is 66, essentially 66%.
Exactly.
Yeah, that's it.
Did you follow Katie?
Yes.
That's a lie. You did not.
I was telling Joe to go
Throw-
switch the
throw your-
laundry. I apologize.
Just you got three that are normal looking
Yeah.
theoretically, and one
I can
that's
do a Punnett
mutant.
square.
Yeah, if those out of those 3, 2 of them theoretically should be heterozygous. But remember that your 1 in 4 and so on is all
Yeah.
just this is what you potentially could get if you do 8,000 crosses.
Yeah. It's all.
Yeah, then you get the
fondest
students just
memory-
copy friends and then remove 2 or 4 from it. That's the problem.
My
One of
fondest
my-
memory. Go ahead. Sorry.
Oh, sorry. I was going to say one of my favorite questions. I was the TA for a breeding genetics class. And one of my favorite questions that the professor wrote for one of the tests was we were talking about calculating, percent chance that something is a carrier for a, non-visual trait. So kind of like what we're talking about. And the question was, how many breedings you need to do? You have to do all this calculating to figure out. Then he's like, and then if you do a cross and out of 10, one of them is visual, then how do you do it? And these students are all worried about doing all these calculations. Like, well, if you get one, then it's a het. But they would freak out. They're like trying to figure out this math. And he asked it after he's asked several other ones, But yeah, if one pops out, then it's a het.
Our daughter is in 7th grade now, but when she was in 5th grade, she had a question on a science test that her teacher marked wrong. And she came home just indignant that it was wrong because it should have been correct. And it was a Punnett square question.
There was.
And so James was like, no, you're right. And we actually sent it to Travis. And Travis was like, no, Joe's right. She got the question right. So then she got to go to her teacher and explain why it should have been. And
This was also the year that I think where she corrected her science teacher over and over again on
Multiple
things her
times.
science teacher was wrong
Multiple
about,
times.
which is the problem with her having a dad who's a science
Yay.
teacher.
Yeah.
How funny.
yeah.
There were so many things like,
She's
well, that's not
rocking
right.
like a 98 in her science class right now, though. I'm pretty proud of her. So
Well, the het thing though is always, I don't know.
she loves Punnett squares. And her response was, well, I know that's what this is because this is how snake genetics work. And I was like, oh my God.
Teach them right. See, and people can not have to worry about heads anymore. Just get a hold of Ben if it's ball pythons, and he can tell you if it's a head or not, and then it can be 100% or 0%, and you're good.
So Tracy said, thank you all for the answers. It's been fun learning all the differences between the different hobbies. In her head, she keeps comparing the two and thinking of them the same.
Birds and reptiles.
They're
I mean,
exactly the same.
I mean, yeah, it's the same across everything. Genetics is genetics is
Yeah.
genetics. It's
Yeah.
just the terminology especially in the hobby, is so bastardized that it doesn't even come close to the way actual scientists talk about this stuff. A lot of the other, as Warren was talking about, the agricultural side of things, and even the people who've picked up like backyard chickens and quails and stuff, so much of the body out there is already in that scientific literature that they picked it up and went that way. The way that hobby got into it was with people who had zero scientific background, zero interest in a scientific background. So they just kind of winged it off of what they remembered from elementary school, which they didn't remember very well.
So that's the other thing about social media that I find funny is, and I know a lot of people, especially that some of y'all try to stay out of it, but I loved when, I think it was Warren commented about Parthenogenesis like a year or two ago, and someone tried to ask him what he knew about Parthenogenesis. Thought it was fucking hilarious.
Yeah.
But that's,
and I will say this about Parthenogenesis: all that work that we're building on has come from from agriculture. It's come from work on turkeys and chickens from the 1960s and 70s. So we're just building upon their work. And in fact, this year it was determined that facultative parthenogenesis, at least in fruit flies, is controlled by 3 different genes, and they have an additive effect. We've seen that work was done 20-something years ago in quail, and it was identified that it was an autosomal recessive trait 50 years ago in domestic turkeys. So we're building on the agricultural genetics. So it's the same thing.
Yeah, I think
chromosomes to chromosomes, DNA is DNA.
Yeah, it all works. It just, some of it, I think, again, get confused for folks because, like you said, the word super, and like I use the word super all the time when I talk about like hypo and boas, and I know it's just a homozygous. I know what it is myself, but if I say that to somebody, they're going to look at me like I'm confused. But
I've got no problem with that. But I got no problem with people using super because it defines what it is. It's a homozygous, as long as they understand that that's what it is. A lot of people think it's different, but they think that a super is different in some shape or form. They don't understand that the incomplete dominant is a heterozygote and the super form is the homozygote. Right. But now you're seeing some people, which is just becoming ridiculous. Things like leopards in boas. Leopard is an incomplete dominant trait, and you call it het leopard and leopard, which is genetically fine. Some people now are calling them leopard and super leopards.
Which is confusing because you got to understand that the het, to them, I guess a leopard is the het, and they don't understand the difference there.
Yeah.
Fine, as long as you het, whatever.
I was going to add to what Warren was saying. I'm all right with people saying super as well, but what I'm not all right with is when they correct me that there is no such thing as a het pastel, because it's a codom. So.
And you're like, so many things wrong in this conversation.
Twice there.
You just want to look at them, nice try, and move on.
Wow. Yeah.
No testing for you.
So for Charles who asked about the het thing, just don't buy hets. Just go ahead and buy visual everything, you'll be good.
Or send it to Ben.
Or send it to Ben.
Some hats
and
can
Ben
be
will
really cool looking. So.
That is true. So that's the one thing I like about incomplete dominant genes and boas. I like jungle and I like hypo.
If you lose hets, you lose normals. So.
Yeah. Well, what's weird about some of the things, I did a bad thing this past year. I bred ball pythons for the first time.
I was like, hell are you fixing to talk about on this show?
That's it. Hold on. Where do I cancel out of this year? Leave.
Hey, you did it too, Warren. You did it too for a while.
Yeah, I know, but I realized the errors in my way.
all I wanted to do was produce a banana. That's all I was trying to do was produce a banana. I was like, I have a banana and I have a non-banana. I got 3 babies, not a single fucking banana. That's all I wanted to do. And now I'm done. I'm like, screw it. It will be different because I will not be doing it. Huh?
Did you get 3 females?
I have not actually sexed them. I'm gonna be really pissed off if they're male and they're not. All 3 are male and they're not banana. That would piss me off even more.
Yeah, that'd be even worse.
That'd be unique.
Well, it depends on what the banana parent was.
Yeah,
it was a male. It was a male because I don't have the kind of money for it. Well, that was given to me anyways, but I still have the money for a female one. Plus, I don't.
Are they not like $25 now, or what are they? $70?
I don't know what they are. I know at one point female bananas were expensive.
I can Google that.
A female banana. I can tell you right now, it costs more than what I would want to spend on a ball python, which is anything.
Chances are good. You got 3 females, so now you got 4 you can breed to that male in a couple of years, 3 years.
There you go.
And still not get bananas.
$6.25 per banana. Whenever we do it.
Well, he's a banana pod, and I was like, I could produce a banana pod, and I was like, she's inchy. I like incomplete dominant genes, so this would be fun. And no, I ended up with like 2 inches and a normal.
This banana het pod is $150 on Morph Market.
Jesus,
They're like,
I
you can't
remember
just find
Daytona
a banana. They're like,
13
it's
years
banana
ago,
like
2 male bananas at 25,000 a piece, and they sold before the show started.
oh yeah,
Yellow bellies. I paid 15,000 for my yellow belly 19 years ago.
Crazy.
Oh,
Never
Oh,
bred
I
from it.
forgot.
I paid 9,000 for a Mojave male once.
God,
I
You
didn't
paid
pay
9
anything for a ball python ever.
Not
I
yet.
haven't
I
either.
know your brain started working on it.
Yeah.
You're just waiting on that next big morph to come in. I know it.
Yeah, you know it.
Yeah, and then what Zach's going to do is he's going to threaten to send people false water cobras if they don't send him ball pythons.
There you go. That's my currency is false water cobras for snakes.
just you got a chart that says how many false water cobras equal how the conversion
I
chart.
traded a lot of false water cobras this year because I hate selling snakes. I don't like it. And I was just like, okay, well, I'd find something I'd want and I'd be like, so how many false water cobras do you want? And one guy jokingly was like, 4. And I was like, done. He was like, really? I was like, yeah, I don't care. Like,
Zach's like, I think
coming
you
out of
underestimate,
everywhere over here.
you underestimate how many false water cobras I have.
Yes. So, yeah,
Wow.
I did that for some. I don't even remember what I think it was a Honduran milk snake or something. But anyway,
Nice. That's
We had someone, we imported we were the first among the first people that imported a zebra jaguar carpet or zebra carpet python. I think this was like 07 or 09, somewhere like that.
From Paul.
And, we had somebody. What's that?
Was that from Paul Harris?
He got one the same. Yeah. It was from Paul. Yeah. So it was us and Nick Mutton and 1 or 2 other people. We all got them the same year. But anyway, we did hatch out some babies. When we got babies, I got a message asking if I would take 2,000 green anoles in trade for one of them.
That's a lot of
Yes.
green anoles.
It ended up being Chris Behoff. If any of you know him, he was just he like made a fake account to send us that message.
I feel like Zach would be like, yeah, I'll take that. That's
Yeah,
At that point, is it
I mean, I got a lot of
food for
animals
stuff?
here that eat anoles. So why
Yeah.
Absolutely, I'm the same. I would take those right now. I got a lot of Trinidad Freebos that need to fed.
Yeah.
I feel like Warren gets that message
I was at
and he gets
Paul's.
pissed off when it's a joke. He's like,
Mhm.
Yeah, just to say, Ben, I was at Paul's house probably in 2001, 2002. Whenever he hatched his first zebra. I
Oh,
saw him as
awesome!
like a tiny baby. It's pretty cool.
Oh yeah. He has some awesome carpets.
Cool animal. Yeah, still does.
let's see. I got some other things here people asked. This one from DGB Exotics says, are there any genetic diseases that you have seen that we think we should test more for as breeders? We should test for. I know, Zach, you probably have to worry about that with people coming in for things outside of the collection. You probably worry about diseases and all. Is there anything that you think we're not testing for enough? I mean, everybody's NIDO, I think, is everything everybody freaks out about, but is there anything else we should worry about?
I know it's not a genetic disease. There's some, this is going to be very vague because I don't know the details exactly. There's some colubrid virus that's kind of raging through some European collections right now that when it shows up, just wipes everybody out. It's kind of like it's not like a colubrid nido, but that's what it's been compared to, because my understanding is that colubrids can carry a version of serpentivirus, but it's not really overly detrimental. And there was some concern because in Europe they just banned king snakes because there's a couple islands over there where the king snakes have escaped and they're pretty gnarly invasive. And so people from Europe were ditching their lamps, and there was a lot of large imports from Europe, back to the United States, because most of those things started here, went over there. And there's a little bit of a concern that with that importation, virus may have come over. But I've just heard like whispers of it. Not that anybody's keeping it on the down low. I don't want to give that impression, but we'll find out in the next year. I can say that.
Well, I can't think of many genetic diseases we worry about. I mean, there are genetic disorders that definitely we try not to breed into stuff that keep getting bred into stuff.
The one that I'm familiar with that people have brought up multiple times, and I've asked for samples, is stargazing in corn snakes. So it sounds like that is a genetic disease. And I would happily, try to find out where it is and be able to help people eliminate that from their collections if they think there's a chance. But I have to have, 50 samples that we know are stargazers, and people don't usually keep those around, so and they try to not make more. So.
Depends on whether it's a genetic disease or whether it's a virus like IBD and BOAs.
Yeah.
One of the arenaviruses.
Because that's what I think about when you're stargazer. I think IBD and BOAs.
So that's where they just stare off into space.
Yeah, they
that's
stare
one of the
up.
symptoms. Yeah.
I have
the one that I'm
vague
most concerned
memories.
about.
I have vague memories that they originally thought that stargazing was associated with one specific morph, and then somebody managed to, for lack of a better term, unlink the two, so that the morph now behaves fine, but you can still get stargazer animals that are like normal and don't carry the morph gene or something, which does have an implication that it is genetically based. But yeah, maybe it just, whoever was doing that had it in their collection and they managed to get the uninfected snakes in the opposite way that everybody else had or something.
The one that I'm most worried about going into collections are snake fungal disease, with people still bringing wild caught animals into their collections. We just saw a grad student giving a presentation a few weeks ago about the range of snakes caught in Virginia that exhibit harbor snake fungal disease, and it is crazy. So pit vipers through colubrids, through natricines, and once people and I know I see on some of these colubrid groups about people bringing in wild caught animals to augment their bloodlines. Once that comes in, they're in a lot of trouble.
And is that something that sits around maybe for a little while? They could actually breed, pass it on, and then it show up in other collections, or is that something that once the animal has it, it's fairly obvious?
They don't know enough about it in that stage, to my knowledge. Gotcha. And they don't have a treatment for it either. Are you aware of any treatment, Zach?
No, I don't think there's any. I found it in the field. It's more common.
I saw it in a copperhead a couple of months ago in Raleigh. I was down visiting friends, and I saw a copperhead. In fact, my daughter or my son saw this copperhead going in front of the trail, and it was really gnarly. It was screwed up with snake fungal disease.
I think that's what happened.
Oh, that's right.
At camp,
so that
it was like last summer or two summers
ago. She sends me a picture of a rat snake, and that's my first thought was it was snake fungal. I mean, because it looked fucked up. I was like, you need to kill that and then burn the body, like get rid of it. And so she did. She disposed of it and killed it.
I did not throw up in the process.
The problem is it's already in the environment. That's the big issue at that point.
Yeah,
you're putting the animal out of misery, but it's already in the environment. So it's.
But I remember seeing that picture of the rat snake. I think that's a nasty looking thing.
Mm-hmm. Yeah, that's what I worry more about. And we don't know. So they included Burmese python. So they sampled from Florida all the way up to Connecticut. So they had Burmese pythons in the sampling from Florida, and none of those had it. Because one of the fears was, where does this thing come from? One of the questions. And a fear was that it came in on imported reptiles, one potentially being the Burmese python. But there was no evidence of it in pythons that they caught. So, and I think they caught like some ball pythons and stuff like that there, but they found no evidence in any of the pythons that they found in Florida. But they also saw a higher prevalence of it the further north they went as well.
Yeah.
So I need to check that. Yeah.
I think it's one of those fungi where, like, as you go south, the heat kind of nukes it a little bit, but when you get to like temperate climates, you're kind of in that magical fungal zone for a lot of fungi.
The magical fungal zone.
You've also got a massive amount of genetic variation. Genetically, it's really diverse. So it's not, it's not beyond the realm of imagination of seeing a variant that arises that is heat tolerant or cold tolerant that we see in lots of other things. Yeah.
And some of it may also be kind of like we see with white nose syndrome in bats, where it's really hits the bats hard during winter when they're hibernating. Mm-hmm. Snakes that are going, torpid and slowing their metabolism down are becoming more susceptible to it. But then, the further south you go, the more active they're able to be, the less likely they're having to slow their metabolism down, the more they're able to battle it off. So that could be accounting for the difference that we're seeing too.
That makes
But
sense. I mean,
also
because the
in
one you
further
found
south
was
you
in
go,
North
they-
Alabama.
Yeah.
There's less
What was that, Warren?
denning behavior, right? There's less of a long-term denning behavior the further south you go because there's a reduced winter phase. So you go up north. I know that the copperheads that we've worked on in Connecticut, they're hibernating for 6 months, 7 months of the year in
Wow.
communal dens with not just copperheads but timber rattlesnakes and colubrids, a bunch of colubrids and so on. So there's the potential at that point because they're not exactly sitting there not moving; they're still active, and therefore potentially spreading this among individuals. So that's it's not a genetic disease, but that's when we talk about what we worry about, that's the one that I'm waiting to see popping up in collections because listening to your podcast, Zach, I've heard some individuals saying that they ruminate or hibernate their females in collectively in groups, or their males in groups, and I think that just increases likelihood that something could spread in time. So that scares me.
That makes sense.
Well, like I said, I think most of the genetic diseases we kind of weed out, and if anything, it's a genetic disorder that like if things like something like JAGs and stuff like that, that's something that we know exists and people keep breeding. But I think most genetic diseases probably get weeded out in collections. It's the true viruses or fungal infections, things that can work their way in and then spread like that are the ones we really worry about.
Yeah, and the other thing is that a lot of these issues, genetic variants that can cause a deleterious effect, we might see very little effect in a single gene, but when you start putting these things together, there's always the potential that you're going to have interactions that you've got. Mutations generally are deleterious in some shape or form. We've got them in somewhat ideal conditions, so it reduces that deleterious effect. But whenever you start adding 5 or 6 or 7 genes together that all have a deleterious effect, the likelihood is you're going to see something popping up from that there as well.
Yeah.
And also, they tend to look like crap whenever you got 6 or 7 genes together anyway.
Yeah.
So it looks like crap, which is genetic, and
yeah, there you go.
So this one is from our friend Jeff Frederick. He asked he'd like to hear the group's thoughts on the current and future use of genetics in speciation and taxonomy, pros, cons, and missing pieces. Taxonomy is always a weird thing because I look at things like how they've split up rat snakes and it makes no sense to me. How they want to lump some things together, and I'm like, I get it genetically, but I mean, if you look at them, they don't look the same.
I think the best thing I've heard said about taxonomists is that they're not going for clarity. They're going for controversy and conflict.
Job
thank you,
security.
Travis.
I'm just telling you that's what I heard.
Yeah, especially those crawfish people.
Yeah, I mean, there's like those crayfish people are assholes. All 3 of us that do this. I don't know. I can speak to it a little bit. My response when I hear people getting mad about rat sticks and everything, just breathe. That's my initial response. Like, it's okay. A species is a hypothesis, so you gotta remember that. You don't have to agree with the hypothesis. However, if you disagree with the hypothesis and you want to stand on scientific grounds, you have to have a counter hypothesis, which isn't that guy sucks. Like, that is not. I mean, I guess technically it could be a hypothesis, but I could say, like, as someone who names crayfish, some of the crayfish I named 8 years ago with new methods like RadSeq and stuff like that. Now we're running those through with the new analysis. We're like, doesn't hold. But if you're a good taxonomist, you simply say, okay, that initial hypothesis was wrong. We're going to sync it, or we're going to merge it, or we got new technology. It was actually 2 things, but that's like all that the people are really doing, if they're honest scientists. So that's my stance on that.
I think one of the ones that was kind of most confusing to the general public was the green tree python thing, right? They showed the DNA difference between green trees, and it was I forget what it was, but it was kind of a large number when you look at the percentage and difference between the 2 different populations, and then you look at the percentage of difference between like chimpanzees and humans, which was such a small number. It was a weird thing to look at green trees and go, yeah, they're a different species.
Yeah, but remember that whenever they're talking about divergence between chimpanzees and humans and so on. They're talking about whole genome differences. Whenever you're looking at a lot of these reptile related, they're looking at a handful of genes, generally a very small number of genes, and they're looking at the percentage variance in those. And you've got a lot of your genome where there's no variation really, or there's you got a bunch of genome where it could be a ton of variation. So it's not necessarily comparable. That 2% between humans and chimpanzees is a terrible example that people use because it's not in the same shape or form that we use for speciation in general. You know, I've worked on animals that are, you know, at some mitochondrial genes they're under 2% divergent, but they are 50 million years separated. Right?
Yeah.
There's just not been a lot of mutation occurring in those genes to result in divergence. You know, it's more significant. And we've done a bunch of work on scrub pythons, and we're going to publish that hopefully this year. That shows some really significant divergence in scrub pythons, which matches the green tree python stuff, which is very clear pattern. There's the thing about the green tree python example is it's not just genetic differences that are significant there. There is phenotypic differences, and there is geographic. There's a geographic explanation for that divergence. And you see that not just in green tree pythons. If you look at any species that exhibits a comparable range in Papua New Guinea, you'll see the same kind of pattern occurring. So scrub pythons, you see somewhat in white-lipped pythons. You'll see it in a whole bunch of lizards, even in birds, because there's a big mountain range in the middle that causes a geographic barrier to dispersal.
Like I know, like timber rattlesnakes are ones that are interesting because their range is so huge that I've heard there's differences in venom between the different areas and all, and but we lump them all in as a timber rattlesnake / canebrakes as the same thing. But I mean that range is massive from Texas to Pennsylvania, so there's definitely got to be some difference there. You would imagine.
There's you go to either range, it's going to be much more different than if you go into the middle.
Yeah,
right? But that's what we expect. Yeah.
Yes. Yeah. And another thing, what Warren was talking about is a lot of taxonomists now, you know, we do what's called an integrated species concept, which is basically exactly what Warren just talked about. You have to have, for me, I have to have morphology, I have to have geography, I have to have genetics. We call it the holy trinity here. And if I have two that are real strong and one is kind of strong, and then it's a judgment call, and I don't like that. And then I'll dig a little deeper and usually find something that merges it or, you know, makes that third example strong when I'm doing the crayfish stuff. The thing that I don't like, the thing that kind of bothers me now with the technology we have today is when species of reptiles are described because there's like a scale difference, but there's no genetics, and there's no the rhino rat snakes were recently split. And that's one that I was kind of like, I was actually excited about it at first. And then I read the paper and was like, okay, there's not enough evidence here to convince me because, you know, I think it was like two scales, the L'Oreal scales or something were different. But I get a little bit upset that in the herpetological world, you know, being a so-called splitter, it has become like you're a blasphemous asshole. And in reality, you're not really, if you're doing your job right, you're kind of looking at if you're looking at multiple lines of evidence and it supports that, I don't see a problem.
So are you more along the lines of putting things as subspecies or complete splitting species?
That goes back to his idea that species is a concept. Yeah. It's not a concrete.
Yeah. Subspecies and me don't jive, but that's like a 3-hour conversation.
I like to tell people that the animals don't care.
They don't.
They don't give a shit.
Well, I think a lot of it gets confused in the what everyone learns is the high school definition of a species. You know, they can't breed, but then you're like, but this can breed, and this can breed, and this can breed, and.
But here's the danger in that. The danger in that is you can take species from Africa and from Australia and put them together and they will breed and they will produce viable offspring. But then you have to take a step back as a just smart person and be like, that's never going to happen in nature. So, you know, I've had so many people come to me when I name a new species of crocodile or whatever. They're like, well, did you put them in the bucket and see them breed? I'm like, no, I didn't.
That seems weird. You know, crawfish porn.
We don't do that yet. A, that's weird. And B, that would cost a lot of money genetically to do. and see, I got enough evidence already.
Just
I don't
gotta
have
love
to do that.
someone challenge people. Did you make them fuck first?
I,
Yeah.
Gotta
I
make
have to
them
go
fuck
pee
first.
at a very
That-
interesting part of
you'd
this
be amazed.
conversation.
There's
The
a
other
little bit
thing-
of biological history though to that, which I teach my classes. There's a guy named Ernst Mayr, and there was a guy named Gaylord Simpson. Gaylord Simpson, best name ever. He was a paleontologist at the American Museum of Natural History, and he kind of fostered this thing called the evolutionary species concept, which talks a lot about how geography matters and phenotype matters, and having your interbreeding uniqueness and all that. It matters, but because geography, that whole the breeding thing wasn't as important. And Ernst Mayr was like the guy that fathered that whole bit where a species is an independent community, blah, blah, blah. And when they were writing the original textbooks for biology back in like the early 1900s, because remember, like evolution just became a thing in 1869-ish, and it took a while for people to be like, yeah, that's a thing. They were adding evolution to textbooks, and Mayr was just so loud about his idea that his idea was the one that kind of got in there, and then was the father, like that was what then everybody was taught. But people don't realize there were competing ideas of what constituted a species all the way back when we first came up with a species. So that's just one idea of what a species is that whole biological species concept thing.
His first name was George.
Wait, Gaylord?
George Gaylord
George
Simpson.
There you go. Well, the people at American Museum that I've talked to refer to him as Gaylord.
Okay.
Oh, how could you not?
How could you not?
the
Amazing name. So,
other thing I like to tell people when they're talking about this subject is you have to realize, like, the scientists that are in these, whether you're talking about, birds or crayfish or snakes or frogs or whatever, like the scientists in that specific group, like they're the ones that have thought the most about the current technology and what people know about these animals from all kinds of different ways. And we kind of have to trust them that it could be kind of like that example Zach just gave. It could be someone in that group just tends to be more of a splitter or more of a lumper, and that could really influence what most people do with that species group, that taxon. It could have a big influence, and 10 years later, maybe there's someone that's the opposite. And so it can kind of change. But the bottom line is, unless you feel like the scientists are doing it so bad in the group that you are so passionate about, if you want to influence that, you've got to learn what they've learned and contribute. And it could be, that you're right. But we have to trust the people that are in those, working with those specific taxa, and know the most about the most, current technology and things going on. We just have to, and to really know whether they're actually splitting too much or lumping too much, you really have to spend years studying.
So
the green tree pythons is a great example of that, because Natouche explicitly said, I'm a lumper, and I'm splitting these. Like, he looked at the data and he said, no, this is plain as day. If it wasn't, he would have just left him lumped, because that's the kind of taxonomist that he is by his own admission. So you have to trust the scientists that, there are some people you don't trust, a certain person in Australia that
say it's one person, yeah.
But
can't say it 3 times or he'll show up.
Well, he's also not really a taxonomist. He's
no.
He's an
He's
artist.
exactly what his last name says.
Yeah.
So according to
Green
four
tree.
different websites, one of Gaylord Simpson's most popular quotes was, I don't know where to put whales. I'm sticking them here, but I don't have any reason for it.
Yep. Taxonomist.
And it was
I
only
was like,
it was
okay,
only,
then.
It was only a
That's
handful
awesome.
of years ago that they split killer whales, right, into multiple
Yeah.
species, subspecies, and that was based on the problem before was that, again, sequencing of a single gene or a couple of genes could tell you one thing, but whenever they sequenced the entire mitogenome of killer whales, they realized that they fell into very distinct different groups. So again, with the technology changing, I was telling Ben a couple of weeks ago when we had lunch, we've moved past, we just sequence entire genomes now. We don't bother with single genes. We don't We just sequence entire genomes. The problem then becomes analyzing the data, because you generate so much data that you can't compute this on a standard computer. You have to use supercomputers, right? And the other thing that I'll chime in about, that in relation to what Travis was saying about the green tree python paper, Daniel didn't do the genetic work on that. That was done by a group in Florida. They're very, very highly skilled in their field, and they didn't just sequence mitochondrial genes. They sequenced hundreds of nuclear genes as well. So 2 different genomes essentially that they used to answer that question, and it was extremely comprehensive. So,
well, and I
that's
imagine
one of the best studies genetically that I've seen in a long, long time.
Well, I imagine single gene studies are tricky because you could look at a single gene in humans and a single gene in crocodiles, and they could be the same, right? I mean, because there's parts of DNA that are the same.
Genes.
Yeah,
but it's mutation rates and so on. I can look at a handful of genes. For example, if we took the data from the amethystine python work that we're doing, I can take a subset of genes that will tell you very little, and I can take a subset of genes that will tell you a lot. But it's the combination of those that will tell you the true story that's going on. So one gene's not enough.
She said amethystine. Growing up, it was always amethystine pythons, and then everybody says scrub pythons now, and I'm like, why does nobody call it an amethystine anymore? It's the name I learned. Like every book growing up, that was
it's too complex.
Yeah,
because amethystine is just one species, and you're dealing with a whole bunch of different species.
Don't make it confusing, Travis.
And you'd want to shorten it down to like just amethyst python, maybe something. You know,
it's an Amy.
It's an Amy python. Amy pythons. Oh.
Yeah. To give a really maybe a helpful thing for Jeff. It was Jeff that asked the taxonomy question, right?
Yeah.
There's a book I read a while ago. I want to say it's called Naming Nature. I don't have it on my bookshelf right now, which is strange. I told you about it, Ben.
Mm-hmm.
I think it's called Naming Nature, and it discusses taxonomy on a much more approachable level than like the high genes and stuff. It can be very helpful for getting people to understand a bit more how taxonomy can be this sort of wibbly wobbly thing that, yeah, it's not absolutely concrete. It's how you look at it anyway. One of the analogies in the book that I really liked was, comparing like soda. How do you break down all the different kinds of soda? Are they all brown sodas? Well, but root beer doesn't taste anything like Coca-Cola, doesn't taste anything like Dr. Pepper. So those are 3 different sodas, but you can't say they're all one soda just because they're brown. Is Mountain Dew and Orange Crush the same because they're both citrus? No, they're very different. How are you going to break those down depends on how you personally do it. I would do it one way, Ben would do it another way, Zach would do it another way, Warren would do it another way. It's where you can come to a consensus overall, looking at the most data that gives you kind of the best breakdown. But again, at the end of the day, it's what I like and I publish and what gets accepted.
What was the name of that book? That sounds
Naming
like from
Nature.
my level. I can read it and understand it.
Naming Nature.
So, all right. This one will be a fun one. Given y'all's background, and what are your thoughts on common names? Because I'm like, it gets so the one that drives me nuts is I always say common names, they're made up anyway. So when people start arguing common names, I'm like, they're made up anyways. You can, as long as we know what we're talking about, we know what we're talking about. It's made up. So growing up, it was red tail boas and true red tails, and so I own red tail boas. And someone tells me my Colombians aren't red tail. I'm like, they've got red tails. I don't know what the fuck to tell you. It's a red tail boa. But I know that I'm not talking about a Suriname. It's not a true red tail. But you'll see people, especially newer in the hobby, fight to the death on that thing and go, you do not have a red tail. I'm like, it's a fuck. We made the names up, guys. Calm down. You know what I'm talking about. So where do you sit on the use of common names, especially like Zach, because I'm sure you use common names inside of names, especially teaching with like the zoo sciences and all that. How confusing does that get a lot of the times?
I make all my herpetology students learn Latin.
Ugh.
Only because if they're going into the zoo field and they want to be a herp keeper, they have to know it, because nobody in the zoo field they're all herp nerds like us. Like, that's why they got that job. So
My 4th grade GT kids are learning Latin right now.
yeah. Oh, and that's the thing about Latin. As a person who teaches like zoology, that people are like, "Oh, scary," and then you just start teaching and you don't let up, and then the next thing you know, it's not as hard to pick up. I make all the organismal kids here take it. Well, they don't. That's not true. I want them to take biomedical terminology, which is a class that just teaches all the Latin roots and like cephalo means head and platy means flat and rhinos means nose, like all that kind of stuff. Because once you learn those roots, Latin names really aren't that awful and they're kind of fun. Like one of the things I'll plug in false waters again. Hydrodynastes gigas is the Latin name for falsies, and it literally translates to giant water monster is what that, that's pretty cool. So you can nerd out on this stuff too.
So see,
we read something the other day, and one of my gifted kids was like, "That has a Latin base of this and da 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 and da 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 and da da da," and I was like, "Okay, let's keep going with what we're learning." Like,
see, I like the idea of learning it that way. I made the mistake when I was in college. I was like, you know what? Because of what I'm going into, I'll take Latin. It was one of the worst classes I ever took because it was taught as conversational Latin.
Uh-huh.
I was like, what the fuck are we teaching?
No.
No one's speaking this.
oh,
And I think I ended up with like a D in that class. I just could not follow.
Yeah.
Like the whole
That's
book
not
was based.
biomedical terminology.
No, the whole textbook was based on like the life of people in Pompeii, and you had like no, this is not what I wanted to learn. I wanted the root to be able to add it to what I already know, and that was a nightmare. It was not what I expected.
But with the common names, there are places that try to standardize the common names. I think it's what's SSAR stand for? The Society for the Study of Amphibians and Reptiles. I think they put out a standardized common name list for herps. Somebody does.
It's the same for a lot of stuff. Yeah. Like Entomological Society of America do the same thing. They have a committee that will every year will go through and determine what is the common name for X, Y, or Z. Yeah.
But ultimately, we use Latin names. I will say one thing that's kind of fun about common names. They weren't considered proper nouns a lot of the time. So that, like, if you said rainbow boa, it would be a lowercase r and a lowercase b, or Brazilian rainbow boa, lowercase, whatever. Now, a lot of those committees that Warren mentioned, realizing these things should be proper nouns, and it's kind of fun because, like, I think with birds, they're proper nouns. It hasn't hit herps yet. It's hit freshwater fish in North America. All of freshwater fish, I think, are proper nouns now. Freshwater mussels are. So when I was writing the book, that was one of the things that Mark O'Shea and I kind of like, because I was like, okay, we're going to make these things proper nouns. And technically, they're not proper nouns now, but inevitably, they're probably going to be. So I wanted to get ahead of the curve. And but some people take that stuff really seriously.
And here's the deal: if you're on social media and there's somebody complaining about something that you're calling a true red tail versus a red tail or whatever, the very simple thing to do is you turn it off and you walk away, and you can actually look at your true red tail or red tail boa and not give about the kid in his basement in his underpants, telling you what you should know. So people get too worked up about this stuff as well. As soon as I see that on these little groups, people try and bring me in on conversations, and I'm like, no, I'm walking away. It's just not worth it. My blood pressure does not need that nonsense. So,
like I said, it's just, that one always amazes me because I'm like, a lot of the stuff if we know what we're talking about, we know what we're like. If you know what I'm talking about, then we can just move on. We know what we're talking about.
No. What is it?
I don't know.
The corn snakes.
What about the scientific name?
No.
Because I still like a Laphicutata guttata, even though I know it's not
No. It has not. Do you really think I know?
No,
any
you don't. But I just like
name?
it's a fun scientific name to say.
Okay, then. No, it's like, is it the Kasachi corns?
Oh, because
or
it's Kasachi corns or whatever the fuck I don't call them.
Castani, is that?
That's Pantherophis lewinskyii.
It's Lewinskyii, yeah.
That's it. That's it.
Because I called them Kasachi corn snakes because I live near Kasachi, so I know that's what I called them. Then everybody else is like, "Well, it's Lewinsky." I was like, "I don't know. It's the same snake." But yeah, I don't know. Common names.
The one thing that I find funny is listening to people talking about Boleyn's pythons, and they're like, "Well, I'm not calling it Somalia. I'm calling it Morelia, because it behaves much more like a Morelia." And I'm like, "Well, I will tell you that I've included Bohling's pythons into the genetic studies of amethystines, and they're nowhere near Morelia. So therefore, the Somalia fits. Just because you don't think it behaves like one in your expert knowledge of having one Bohling's python or maybe seen one once in a zoo, doesn't mean that, that because you think it is, doesn't mean it's right. People have put a lot of effort into studying these things. So
let's look at the corn snakes. I know the reason for changing the scientific name. They don't belong in the same group as all the rest of the corn snakes or all the rest of the rat snakes on the other side of the world. It's just fun. I grew up on a Laffy Guttata, and it's fun to say. And it took me a while to figure out how to say Pantherophis.
All the tarantula people use Latin names too.
They're fucking weird,
if
though.
the paper you're reading or the species that you're looking at ends in Hoser or Hoseri or my dog Hoseri or whatever, just ignore that. That's not real. And just go back to what it should be.
Good to know. Good to know.
And that is one thing that I think a lot of people don't realize. When you're in academics, you come across it, but if you're not, you may not. Just because something gets published in a scientific journal, not all scientific journals are the same. And some of them don't require very much or any editing or verifying.
Or review.
Yeah.
You can just pay them and they'll print it.
Tell us how you feel, guys. Come on.
Yeah.
So yeah, just because it's in a scientific
All of you at once.
It's
Peer
all over
review.
sciences. There are just some seriously shit journals out there that you pay to get your article in, and now you're the authority on this thing because it was published, but it's bullshit science that you paid to have promoted.
Hey, if they put science in the title of the journal, it's got to be good.
Oh yeah,
God,
sure.
James.
There's a whole religion about that too. It's called Scientology. It's so legit.
I think you've got to pay to be in that as well. So.
Yeah, you do.
Yes, you do.
Oh,
and
man.
Scientists in whatever area it is that you publish that bullshit stuff, no, it's bullshit. But if you're marketing to people that aren't scientists, then they'll just believe it because it's in a scientific journal.
But I read it online. It has to be true.
You know how many times I've had to teach kids mermaids aren't real?
Oh,
Like, I'd like to say I'm joking, but all the time I have to teach them that mermaids aren't
I
real.
teach 9 and 10-year-olds. So
Teach high
we
school
believe
kids.
in a lot of stuff in my class.
Here's what I suppose you're doing the same thing with Santa Claus. You bastard. Yeah, I know the kind of guy that you are.
Hey,
This house believes.
that is real. That one's real. Mermaids not, though. Mermaids never brought me a
All
present.
manatees.
Oh, let's see. Here's another question. Are there species-specific frequencies for morphs spontaneously popping up? As a random example, is albinism more likely to spontaneously develop in a tree boa versus a green tree python? Are there any known environmental or genetic cues that favor occurrences of new mutations?
Mutation, yes. Radiation. Anything, certain chemicals. So you use insecticides and stuff like that. They can be genotoxic and they can cause mutations to arise at a higher frequency. And ionizing radiation can cause it. But in terms of is species A more likely to mutate more than B? The answer is no. The reason you'll see it more in ball pythons or corn snakes is because they're simply bred in higher numbers.
Yeah,
the more
population size helps.
If we know that a mutation rate is X when you have an individual, when you have 2 individuals, well, you've just doubled that because there's 2 chances of that happening. And if you do that by 4, you've just increased it again. So the more individuals you are, the more likely you are of seeing a mutation arise. Because we've got 8.5 billion ball pythons currently on morph market means you're going to see a potential of seeing a lot of mutations. Because you have a lot of corn snakes, there's a potential to see a lot of mutations, but not a lot of people are breeding emerald tree boas. And therefore, you have to reduce that right the way down. So what's the likelihood of seeing that mutation arise? And it's just, it reduces incredibly. Are there albino emeralds? Yes, there has been a picture of an albino emerald tree boa before and a picture of an albino Amazon tree boa before and stuff. But just because there's much fewer individuals, you're less likely to see it. Just it's a numbers game.
The trick is you got to hide it inside of a pregnancy belly and sneak it across state lines and then bring it to America.
Hey, I read that book. It didn't end well for
and then
them.
put it on the cover of Reptiles magazine and pretend that the albino animal that. Anyways, it's one of my favorite ones.
And then breed that to a bunch of other species that are different species, and then tell people that it also arose in a different species from a different locality because you knew exactly what to look for. Yeah.
Yes.
Yeah. And the weird thing is, I've got 2 of those in my collection.
So I know this is Amanda ready and not Lee ready because she's talking about cockroaches. And there's no possible way that there's a scientific name for a cockroach that ends in magnifica?
Totally.
Oh,
is.
yes,
yeah.
there is.
What?
Yeah.
There's tons of animals
There's
that
a
have
couple
that
of them.
outside. That's a common scientific
Because
name.
in my mind, I automatically go magnificent, and there's nothing magnificent about cockroaches.
You haven't seen all the cockroaches yet.
Yeah, I was gonna say you haven't
See,
seen
the
those.
one she's talking
really
about is
good.
pretty.
Really good
All right, let me
cockroaches.
look it up.
Emerald cockroaches
Let me look it
are
up.
pretty.
Let me look it up.
There's even the German cockroaches. They're the shitty ones you don't want. We've got a stock of German cockroaches in the university that are all these different, like red-eyed forms, and winged forms, all these different variants. Some of them are really cool. I should start selling them at.
So Warren's gonna start
Yeah, there you
selling
go.
more
This is
stuff.
the new
There you
isopod.
go.
Maybe
There
we
you
should
go.
breed. Don't
That
worry.
would be
doesn't
awesome.
even
That's a
look
good
like
Just
idea.
a
add it to your
cockroach.
garbage. You'll have 20,000 in a day.
Okay, that looks like a fancy isopod.
Well, it's a type of. I mean, it's a beetle. It's.
So
They
there's
don't all
the,
look the same.
there's
Yeah,
the
cockroach is
emerald
a beetle.
cockroach, and then there's the cockroach wasp.
Yeah, the cockroach
Yeah, they're
wasp is not
different.
it.
They are different. That looks like a fancy
Last name,
isopod.
too.
That doesn't look like a cockroach.
But that's. I mean, look at a German cockroach and look at a hissing cockroach. They don't look alike.
No, they're both gross. This is not gross.
Hissing cockroaches
Some
aren't
of those
gross.
cockroaches that don't have wings, it's the wings that tend to creep people out.
They
I
are
think.
gross.
Yeah.
I mean, it
Yeah.
depends.
But, like,
I
there's
keep
one in-
domino
I know-
roaches, and they're awesome.
Have you seen the one when you were in Costa Rica, Warren? That's like it's a big honking bastard, and when it's walking through the
Oh, that's cute.
leaves, you can hear it. It sounds like a flipping lizard or something, and it's a
Yeah, I think I've got a
Looks like
couple
an
of pictures on my
isopod.
phone from my last trip.
What's
there.
it called?
Yeah, they're huge.
I don't know its name. It's just a really
Just a
cool
big ass
bug.
cockroach. Yeah.
Yeah.
Search big
There
ass
you go, big ass
cockroach.
cockroach.
Let's see.
But there's some
Big ass
cool ones, like look, but whenever we're doing night hikes in the rainforests, the cockroaches are the ones sitting up on the tree trunk or on the branch,
Mhm.
sitting there. Some of them look really cool.
Well, I've learned in the recent month
Do I want to
that
keep them?
I've learned do not have cardboard boxes in your garage.
Okay, but this domino
That's-
cockroach doesn't look like a cockroach. It doesn't look like what my mind thinks of. As
Well, maybe
a
you should see more cockroaches,
Do you like spiders?
They don't bother me.
but they
Right.
don't look the
Do
same.
you like scorpions?
They don't bother me.
Right.
I don't know why
despise them both.
cockroaches bother me.
I don't like them, but a cockroach doesn't bother me.
Yeah.
Do I want to have one in my house? No. But
Oh,
I
we-
can still appreciate that they're kind of really interesting
I
individuals.
used an entire
We-
can of
species.
Raid in my garage.
So we cleaned out our
See, you're
garage
the
over
one that's
Thanksgiving
causing the
break.
insecticide resistance.
is.
Yes,
There you go.
yes,
is.
it was me.
No, look,
Your
we stepped on a lot.
falsification of your genetic data and your
We-
use of RAID.
We stepped
Yeah,
on a
stepped
lot.
I stepped
We
on-
didn't step on a lot. I stepped on
I
a
stepped
lot.
on some.
You stepped on
I don't see
two
roaches.
in the whole day.
But
Don't even.
that's when I found out apparently cockroaches eat cardboard, and we no longer have cardboard boxes in our garage.
Nope.
Well, if you
I
look,
I
the
do
way
have
we raise
kayaks there, though.
the way we raise German roaches in my animal room is we use cardboard boxes sheets of cardboard on top of each other because they'll all hide in between them. So they don't really
Yep.
eat it,
Yeah.
but they'll use it for
We found that out the
Recovery.
hard
We
way.
we had our garage had they weren't the Germans, they were the with the wood roaches, American roaches, whatever. We had tons of them, and so we started cleaning. I'm like, there's got to be something they're eating, and we got to the box where they were all at, and it was just full of like DVDs. There was no food, no nothing. I'm like, what the fuck are they? But
They're eating DVDs.
they're just eating DVD. Found the use for DVDs.
Pretty sure
They
I
don't
had
know that
PTSD
they
after
need a
that.
TV to watch the DVDs. They're just
Yeah.
watching DVDs.
Just
Yeah,
watching TV.
we're going
And if
to
you find
Joe's
any of those-
apartment territory now.
Yeah. And if you find any of those American roaches, just send them down to Florida because there's a shop there that does like cockroach eating competitions, right? Or was that a
Oof.
bunch of years ago?
That
Oh.
was
He
that
died
was,
from it,
that was
right?
Ben Siegel.
Siegel.
Oh, I remember that.
Yeah.
The Alicia, to answer Alicia's question, the big ass cockroach Latin is Balaberus giganteus, and that's the one that they were talking about.
See, now
And
you
I
know.
actually
to answer
Googled
Alicia's-
giant
to answer
Costa
Alicia's
Rican
question,
cockroach.
Yes, Alicia, you should breed more hognoses. I've been to Alicia's house a couple of times, maybe once.
Breed more
She's got
hognose.
some really, really cool hognose snakes.
So here's a question that it's as someone who I do obviously do not understand genetics at the level y'all do, but I do understand genetics. So every time I hear somebody talk about inbreeding, my mind goes. My mind's always like, it's not that bad, but then you have to explain why it's not that bad. And it's really weird. So
Wait. Inbreeding and what?
in general,
No.
it's genetically,
With animals?
yeah. If there's nothing that no negative. Anyways, I'll let them
No
explain.
people.
So,
These
I'm
Southerners.
not-
Yeah.
Hey now.
so Lee already said the effects of inbreeding in reptiles, because you always hear about you shouldn't line breed or inbreed.
I don't have inbreeding in my family tree. Somebody else does.
Whoa. not me. That is
Family
someone.
stick. There's no branches.
No,
Yes.
I'm in a different tree. That part of the tree is growing in a different yard. That
Yes.
is not this tree.
We cut that part of the tree off.
That is not. All of my blood is not related.
Wow, that really hit a point there, didn't it?
Yeah, it did.
Wow.
To make everybody clear, that is not me.
I can tell you my favorite saying about inbreeding. I had a student. He's just this farm kid, just total farm kid in this breeding genetics class we were in. He's like, well, what I always figured out is if you're breeding related individuals together and it turns out good, it's line breeding. If it turns out bad, it's inbreeding. I was like, you know what? You're
That's
right.
That's pretty much the long and the short of it right there.
Oh, that's funny.
So I'll let. Which doctor wants to explain issues or non-issues with inbreeding?
So inbreeding, I work on organisms that are highly inbred. So like when you work with bed bugs, they're almost within populations, they're almost clonal because there's literally no genetic variation in the population because they are founded from highly inbred populations. They are introduced into new areas and they inbreed among each other, and then you lose variation. Now, inbreeding is not necessarily a bad thing because inbreeding has been shown to also increase the rate of the removal of deleterious mutations. However, inbreeding can also fix deleterious mutations depending on how deleterious that mutation is. So if it's very mildly deleterious, very mildly, bad, it can get fixed in a population. And as you accrue those, it can end up being one that's really bad, and that can cause the whole line to go extinct, which is what you see happening in mice breeding populations, for example. People working with house mice, they line breed and line breed and line breed. And as they accrue mutations, eventually one thing will kick them off and they're destroyed. The problem is you can also fix those through line breeding, and then you can outbreed them. And when you outbreed them, you get outbreeding depression, and it can be just as bad because you're breaking up gene complexes that were good. You're introducing deleterious mutations, and that can be bad. So line breeding and inbreeding are the exact same thing because you're breeding individuals that are highly related to each other. And Ben is exactly right. Whenever it's good, it's line breeding, and whenever it's bad, it's inbreeding.
So,
what I will say is that I also find people that want to have their name associated to something line breed, and it's their line, which I think is complete nonsense. I work with a lot of Hog Island boas, and you've got the Shuet line, and you've got the Sears line, and this and that. I don't line breeders. I cross them all because I want to maximize genetic variation among them. So if people want a Sears line from me, they're not going to get it, right? If they want a genetically pure Hog Island boa, they'll definitely get a really good one. You'll also find there's animals that we can't do anything but line breed or inbreed. A lot of these very rare island boas, the K cockers, the West Snake K, Lagoon K, they started from a very, very small population, and there are no more individuals to collect. Essentially, you can't get them. So there's been a lot of inbreeding going on there, which has caused these deleterious mutations to be excluded or removed from those individuals. You can't do anything about that. However, with your ball pythons and your boa constrictors and so on, there's no reason why you cannot outbreed those to different lines to increase genetic diversity. And all of the sequencing, I don't know what Ben would say from his stuff, but in all of the sequencing work that we've done on ball pythons, boas, and a handful of other hobby-related snakes, they lack genetic variation in a big way in compared to a lot of the wild populations we work with, because there's just been a lot of line breeding. Do I support it? I have inbred the shit out of a lot of stuff, but then I also outbreed. Once I get what I want, I then outbreed it.
Yeah.
To then strengthen those lines. It's just being able to do that.
And going along with what you're talking about there, the difference between inbreeding and line breeding. The hobby doesn't really do line breeding per se, because what line breeding means is if you get something deleterious, you purge that out of your collection. And the way we purge things out of the collection is we just sell it to somebody else So those bad genes stick around in the collection. In agriculture and stuff, when they purge that out of the collection, they go out and they sterilize that animal, and it doesn't get to breed anymore. And they will go so far as to not only does that animal not get to breed, but none of its children get to breed, and none of its parents breed again either.
But Travis, that snake cost that person $3,000. You can't expect them just to get rid of it.
The same thing happens in the dog world. But the reason it works there for line breeding works is because they keep really strict pedigrees.
Yes.
Dog world, I found it. So I worked at PetSmart for a while.
Well, that's a different. PetSmart dogs are a bit different than.
Well, no. I would learn things from people that would come in, and someone brought in a white boxer. I'd never seen a white boxer, and it was blind and deaf and had all these issues. And they just like, yeah, they tend to be born with all these issues, but apparently, a lot of breeders will just kill them all when they're born, and then not tell people that their dog produced white boxers.
It's also not true. That there's a whole misnomer. White boxers are not more likely to be blind. I had a white boxer for 13 years, and it turned out to be a very healthy animal. I'm talking to other people that had white boxers. They had no issues with them. Just like any other boxer, they can be blind or deaf or whatever. And in fact, if you go back and look in the original bloodlines, white boxers were actually very prominent in the early years of fixing that lineage. And it's just what fits in at that point with the Kennel Club. But as I was saying, in the snake world, not a lot of people keep very good pedigrees. You see it every day on Facebook and on Instagram or whatever. They bought a snake and they're asking questions. You're like, who'd you get it from? And they're like, Steve at Snepticon, and they don't know who that was, right? And they don't know what their animal is. So if you buy from a breeder, they should be able to tell you at least the last several generations of that animal's lineage. And if you can't get that, then there's a question behind those individuals.
Well, and also I think more people need to, if you're buying a reptile for a certain gene, if it's not one of those genes that is like only this one person has bred it and this one you get it, you don't always have to buy the pair from the same person, right? You can buy one from this person and buy one from that person. It's a better chance of them. One, if you buy a pair from somebody, it's most likely a brother and sister from the same clutch or same litter. I like the idea of a diverse gene line and all that, but yeah, it gets very, like I said, people hear inbreeding, and because of the way it gets put on TV shows and cartoons with people, they always go, well, if we inbreed, it grows 3 arms. I'm like, that's not how inbreeding works. That's not how any of this works. It doesn't magically grow another appendage. It's not. So it's a hard one to explain.
That we're currently eating, whether it's vegetable or animal, has been inbred to select for traits. The difference is that it's then been outbred to then help fix traits and remove those that are deleterious. So I don't recommend it in the human world, but I certainly don't have a problem with it in the animal world. And with snakes, as long as you don't continually inbreed, I don't see a problem with it. But then you look at someone like, I think it was Ron Tremper. I think he inbred leopard gecko lines for something like 26 lines or 28 generations, something like that there. And what you tend to find is that once you get to about generation 6 or 8, you start seeing fixation of mutations and start seeing visual variants arising. So if you look back at Ron's work, I think it was either in the 6th or the 8th generation, you started seeing these high yellows and stripes and stuff like that popping up among his collection because they've been inbred, and you're fixing the these heterozygous into homozygous, and you're more likely to see that variant.
I think for a lot of traits, when people line breed that they're breeding for a certain look, you can probably find a similar look in a non-related individual. So again, breed for what you want. If you get that animal, then go find something that is close enough but not related, and breed it into that, and strengthen up that gene pool there. And you can still get to that look you're going for.
Remember also that sometimes the same phenotype could be controlled by different genes, and therefore you could actually be causing more problems by selecting to breed it to something that looks similar but is unrelated. So my recommendation to everybody is, it's terrible to see what's happened to the ball python world, in that a wild-caught import ball python is worth essentially zero. And yet those, in my opinion, are some of the most genetically valuable animals on the planet, because once you get your really cool animal, you breed it to those, because then you've got a really nice genetic background to breed into and then select from. But nobody wants to do that because they don't want ordinary looking ball pythons in their collection. For my stuff, I always outbreed the wild type animals. after I've inbred just to fix traits in homozygous forms, I'll outbreed to wild type locality animals or wild type animals that are nice just to be able to strengthen the lines. So I'm not advocating inbreeding, but it's used to select traits. But I'm advocating outbreeding once you've selected your traits and you're then able to, finesse that lineage over time.
Yeah, he's not advocating inbreeding; he's advocating line breeding. It's much better.
That's right.
And if you don't need to do it, then don't do it.
Though,
yeah.
I one of the things that drives me crazy in herpetoculture is there's the culture of herpetoculture. So, like, there's some species of snake, like diamond pythons. I think I've listened to enough NPR, even though I'm not a Morelia person, to know there's the San Diego Zoo line, and then I always say Sedgwick. That's not right. Riverbanks, is that it? South Carolina. And isn't it that the ones that got to Riverbanks were born in San Diego, and they're all basically the same? So we have this culture of, like, they're literally siblings. I think I'm right in this, but they were released to the public from two different locations across the country, but it doesn't matter because genetically they're all essentially the same. Like, I think it's
I saw that with Brettles.
Yeah. I think we need to question
all of these different lines. Just a bunch of them, they're like, they're all pretty much identical.
How is it that Stonewash spontaneously showed up in three separate lines of Brettles? Well, it's because they're all the same damn line of Brettles.
So I think it's important.
I know several people weren't happy with me releasing loose data, but it is what it is. Yeah.
I don't think they were.
But you're right.
Anyway.
Yeah, you're right in that. But it's just that naming a line, and those zoos might not have specifically named the line. It's the people they got them from.
Yes.
They got it from the and then they maintained that. Yeah.
Well, that's how a lot of morphs are. I mean, a lot of these morphs come in either from one snake and then spread out to multiple collections, and then people. That's what drove me nuts about ball pythons was the naming of morphs, right? Banana and coral glow. It's the same fucking thing. It had to have come from similar. It's the same thing, but because someone on one side of the country named it this and someone on the other side named it this, now we're two different things. That part drives me nuts, along with 5 million other things about ball pythons and the people that breed them.
Yeah, but I don't have an issue with that. In that, if they came from different origins, if they came in from the same animal that then bred, and then somebody decided to call theirs coral glow and somebody else called theirs banana, then there's a problem. But if they were imported from different areas and they were phenotypically similar, I'm happy to call them different things until they're genetically proven to be the same mutation. And what I mean by the same mutation is, I mean the same mutation in the same gene. So the same base pair mutating in the same gene, then I'm happy to call them the same thing. But I think it's very valuable to have different variants within a mutation. I think it's very important to have leopard boas, onyx boas, and Costa Rican black boas because they're likely to be different genetic mutations within the same gene.
Yeah.
And I think that's important. So I don't have a problem with that. I think having, as long as it's coming from a wild origin, then I think it's fine. The difference is if it was from the ball python breeder in Africa, Noah, if he had that original coral glow and he sold a bunch of them, and then he said, you know what, I think I can get a bit more money here by calling this one banana, but it's from the same parents, then I think there's a problem there.
Well, I mean, that's basically what happened with the candy and the toffee. It wasn't from Noah, but both of those animals came in to Outback as siblings, and one got sold to God, I can't remember who, the guy up in Canada, and the other one got sold to somebody here, and they called them different things. They are siblings. I have talked to the guys at Outback. They are siblings, but there are people out there who insist that candy and toffee are two different things. It's the same thing, and Ben can absolutely confirm that.
Dr. Seidel did that work. Yeah, they're identical. So
I love the panic you threw through ball python worlds when you started realizing that things are the same thing, even though people have said it for like forever that this is the same thing as that. People's brains just melted. Like, it can't be because I paid $2,000 more for my version.
Yeah. Yeah, and I think there's sometimes that people don't want to send sheds for certain projects to develop a new test because they don't want to know for sure if it's different or not.
That drives me nuts. That part drives me nuts.
Thankfully, there's enough people working with most genes that even if some people don't want it, other people do. So we'll get there eventually. But yeah, it's interesting. I'm much more science and animal loving than I am a business person.
Yeah.
And so a lot of those practices and philosophies were, kind of jaw dropping for me, but I've gotten more used to it. But I don't sit and think about something like that from like a business perspective. That's like one of the last things I would ever think of it from. But there's definitely people that do.
So one of the last things I want to talk about is the hobby with y'all, because y'all are all kind of at different parts in the hobby. Warren's trying to stay out of it, away from people in general, in his own world. But like
in general, but for me,
like Ben, in recent history, you've kind of, I feel like, you've got thrown more into the hobby because of what you do now. How do you feel where it's at right now as far as a whole? Like compared, I mean, all of y'all have been in the hobby. I want to say a long time, but we've all been in the hobby a long time, right? And it's definitely changed in the last 20 years. Where do you feel now? Is it better, worse? Are we ruining it and it's going to fall into the ocean one day and die?
For me personally, the biggest difference by far was the internet. Like I remember before the internet, and I love that, a 6-year-old can study about, how to keep or breed something or what something's doing in the, you can look up natural history stuff about various species, and you can read, all kinds of stuff from people that, go and have experiences in the wild or keep things or this or that. I think that's awesome. To me, I've seen these, like, as far as the economic side of it, I've seen these, boom and bust things multiple times over the last 20 years that, that part of it doesn't bother me. I know there's a lot of people, not only reptiles, but other pets, during COVID, they got a cat or got a dog or got some geckos or, a snake or whatever. So there's a lot of new people because of COVID years. That, maybe they're producing their first babies right now. And so they're going through their first time seeing an economic downturn. I don't know. To me, I guess seeing those cycles multiple times, it's hard for me to get too worried. I think that a lot of people use up a lot of their energy thinking about and worrying about that kind of stuff. If they're animals, if you're choosing to keep animals that you like and you're interested in and love, if prices go down and you think that it's not worth hatching out more, then just don't pair them and still keep the few that you have and enjoy them. And if prices come back up, breed them again. I don't know. I also have never bred to be able to pay my bills. I've never done it as like a full-time job. But I see definitely the access to information is exponentially different than when I started. But the people that really love it. What's that?
So it's not always good, that access to information, unfortunately, because
yeah. Well, there's a lot of
people don't know how to Google properly.
Yeah. You definitely need to be careful and not just believe everything like we talked about earlier. But yeah, I mean, if it's something like, for me, for my mental well-being, and really it wasn't till COVID that I fully realized, how good it is for my mental health to go into the snake room and just hang out in there, see who's doing what, and look at, potential future pairings and think about that, and look at an animal that's growing up and, remember, just all that kind of stuff is really, for whatever reason, for me, that's very healthy and uplifting and exciting. And, if I don't pair, like these last couple of years, because of the new business, we've been super busy. I had 2 years ago, I had almost 70 clutches. Last year I had 11. And this year I haven't paired anything at all because I just know I'm a lot more busy, but I'm not getting rid of my breeders. I still have them. And, eventually I'll probably start pairing a bunch more again, but it's going to change over the years, and that's just how it goes.
So Zach,
I think if you're keeping it for the right reason, You know, I think it's if you're keeping it for the right reason, you're keeping it because you're really interested in them. And I think
Yeah.
where the hobby's really let itself down in the last bunch of years is that people see money attached to animals instead of animals
Yeah.
attached to animals, and you know, wanting to learn about their natural history and so on. Like I will say that I think the general understanding of Mendelian genetics is exponentially better now than it was 20 years ago in the hobby, and I think that's really a good thing. And it's not necessarily coming from their educational background, but it's coming from their interest in understanding what happens whenever A and B breed and produce, you know, whatever. So I think that's really beneficial. I think, as I say, where the problem arises is whenever somebody just looks at this and thinks, I'm going to make a ton of money from buying all these animals. But I think the hobby also self regulates, because 3 or 4 or 5 years down the line, whenever they're you know, they can't afford to pay their rent anymore because they're having to spend $1,000 a month on rodents, and they sell their collection, well, they're not getting back into it in most cases. I think a good thing for the hobby is you're also seeing diversification happening. So people are keeping multiple animals instead of being a monoculture.
Yeah.
Generally, monoculture is not great because you lose those little, you know, differences that are occurring between species. You know, I keep 130, 140 animals, but a bunch of whole different species. You know, pythons, colubras, and boas, and they're little variants that occur between them in their behaviors and so on. Really, I find really cool and really exciting. And if I didn't breed them, that's not going to bother me because they're still cool animals to keep. As long as you can afford to keep them and feed them, then it's great. But Ben's right. You know, the amount of information that we now have available at the tips of our fingers is incredible. And as long as people are doing that because they really like the animals and not because they like the theoretical dollar bill that comes associated with it, then, you know, I think the hobby's in a great place. I think the hobby's in a better place now than it ever was. And even as much as I'm not a fan of the whole ball python hobby, because I look at more as a pyramid scheme than anything, and less of an actual interest in animals. I will say that they've been a good gateway drug for many people to then get into other species. So I'm happy to support that as well.
And I've said that about I've said it before. I don't hate ball pythons as a species.
No,
and I definitely
animals.
I definitely appreciate most of the advancements we have in the technology and stuff we have in the hobby is because that part of the hobby exists. It's just the people part that really starts to bother me. Just seeing a lot of, I hate saying ball python people, but it's certain types of ball python people, and you've all met them. They're fucking annoying. But I always tell folks when they get it, if they're, you know, if it's a ball python person, the breed just ball pythons, like try keeping something else. Because a lot of those people haven't kept a different species. They don't know that there may be another species they like even more than the ball python that they're currently breeding. And so I like them trying something else because they start to get into some of the other, say, odd snakes out there or other reptiles out there so that they don't disappear from the hobby. You know, I love rubber boas. I wish more people had rubber boas so that it was easier to find rubber boas when I want to buy a rubber boa. Right. So yes, I like the diversity. I'm starting to see more and more people get, you know, some weird type of Asian rat snake, or, you know, the false water cobras or something that is different than the 4 or 5 main snakes that we sell in the hobby. But so I want to ask Zach the same thing about the hobby, but I want to ask you, do you find, because of where the hobby is now, does it help your teaching the zoo science stuff, or do you have to correct a lot of issues? Like, do they come into you
That's a good question.
going, I know how to take care of this, or
I get the gamut, but the thing that I do get, which is cool, the majority is I'm here to learn. Like I don't really get a lot of like, I'm not making, I don't know what else to say. I don't get the ball python bros.
that's the ones that,
but I don't know if they're necessarily coming to college. So, you know, but I get the nerdy kid is the kind of archetype zoocide kid, which I was back in the day, but they come in and, you know, they want to learn. They like to learning something that they're into. And like Ben said, first, there's so much out there for this generation, and for any generation that's entering herpetoculture now. You do have to acquire a little bit of a skill set to be able to weed out the bad information, the good information. I spend a lot of time with the undergrads and even grad students, just trying to get them to understand, like, if just because you get a Google hit doesn't mean that it's right. You got to learn how to vet. I in my herpetology and herpetoculture class, I make them do these things called taxon reports, which is basically these massive lit reviews of any species they want. They pick their taxa. So if I've got someone that's kept bearded dragon, I had a student this past fall who kept bearded dragons his entire life, like literally was born into a bearded dragon. His father bred bearded dragons, I believe. So beardies were there the whole time. And he just was nerding out on the bearded dragons. But even him with that background, I was kind of like, now hold on a minute there, bud, because some of these citations, like, you know more than the person that wrote this. And you should have read that and realized you know more than don't just cite it. So I think that's the part of this whole thing that's been fun for me. And I think that the good breeders, the good herpetoculture peeps, the people that just want to keep snakes and have fun with it, you have to acquire the skill. I think that there's at times a little bit too much tribalism. When I started doing podcasts back in the day and everything, I would talk a lot about like naturalistic keeping. Okay. Because I like to keep snakes in boxes with sticks. Like that's just the way that I like to do it. Because I like to look at snakes on sticks. Like that's it. But I think that it was interpreted like we must keep them with the sticks. And if you don't do that, you're an asshole. And I was just kind of like, what the hell? And then I started hearing my name being used, like Dr. Loughman says. And I'm like, whoa, first it's Zac. And I just like snakes on sticks. Like, let's calm down. So there's another side to it. Like the ball python peeps are getting a lot, but there's that other like holier than thou approach to this whole thing that to me is a little bit off-putting. So in the end, I think all of us here, we just keep the way we want to keep because we like to keep.
Yeah.
I like maybe our letters behind our name gave us something, but I don't think it really. I didn't get a PhD to keep snakes. I got it to actually keep my job. That's why I got it. So but I like where things are going. But I haven't gone to Tinley. I've only been to 2 Tinleys. Warren was at the Tinley with me this fall. And I talked about this on my podcast. I was overwhelmed by the humanity in the room. Like I was so used to growing up in the 90s, early 2000s. Like I was the only kid that did this in my circles. There was no one else. Like I got Reptiles magazine in the mail. And for about a week and a half, it took me to read the entire thing twice. I was in my reptile nerdy world, and then I was in a void. And now I have my herp nerds are over there. That's a term of endearment, by the way. And they're having debates and things, and they're talking about what they saw, here, what they read there, and it's all, so it's just what you make of it's what I'm trying to say. Like you can get caught up in all the drama and the bullshit, or you can basically be like skim the goodness off the top and just stay there. But you determine what you ingest. So
yeah,
and if you do that, it's good.
Yeah, you get out of it what you put into it.
Yeah.
And if it's causing your blood pressure to increase, you're in it in the wrong way.
Right.
Yes.
Get back.
Yeah. But one of the things I think that's really cool is being that I am a data-driven human, for our podcast, Colubrid Radio, shameful plug, Eric gave me password information so I can actually go in and look at like the data and the response to the episodes. And one of the things that makes my heart go pitter patter about it is the episodes that actually are getting the most traction are things like when we had Travis on and talked about kukri snakes, or when we had somebody on to talk about garter snakes. When I did the false water cobra episode, like the obscure animals seem to be getting the most traction, which is nice because when we do corn snakes, we get traction, but we don't get the same traction as Oligodon.
Well, I like having weird. We had was it April Linkfield on, and we talked about like
we had her
snakes
on.
and house snakes. Yeah,
yep. Hers went through the roof. So I don't know if that's just our listenership, because you got to kind of look at this devil's advocate it, and we got the Colubrid nerds are the ones that are listening to us, or if it's literally like people are starved for something different. And so they see that and they're like, oh, I'm going to grab onto it. So I think that we're going to start to see the diversification. I know just in the first year I went to Tinley and this year going to Tinley, I saw more diversity. I don't know, but that's a sample size of one. You know what I mean?
Yeah.
But
Zach, I'm gonna say one thing here. You mentioned about people being excited about the diversity. One thing I will also say, being involved in the podcast, Boas on the Morelia Pipes
there we
Radio.
go.
Kind of group.
Yeah.
What at Tinley and in other places, people came up to me and said they really liked hearing a podcast that links science and natural
Yes.
history and keeping.
Yeah.
It's not what they don't want are I keep my snake in a Freedom Breeder 70 on X substrate, and I feed it every 10 days on X rodent,
Yes.
and I cool it too. That's not interesting to them because realistically, I've got 12 different species downstairs from Sanzinia through to whatever, and I keep them all exactly the same. And guess what? They all breed and they all eat. So there's not a lot of variation that I can talk about. But people like to hear about the natural history
Yep.
and their variation and their distribution and so on. So, and you know what? That's a good thing because that's taking it away from I'm into this for the dollar bills that I can make.
Exactly.
And
Yeah.
I'm into it for understanding more about the species that I'm keeping or that I might want to keep. And also, if you've got a rack, I'm not a bioactive person. I don't keep anything bioactive. But I will say, even if you've got a rack, you can still make the cage look pretty interesting to an animal in terms of
You can.
adding structure to it that allows the animal to get excited about being not just in a box that's a Freedom Breeder 70 or whatever. My Dunn's pythons are in Freedom Breeder 70s or CB 70s, whatever they call them. But there's so much branches and different substrates and highs, so on, that there's a really complex faunarium, terrarium, whatever we want to call it, for them to move around in. They're not just sitting in a blank box. So because you've got a rack doesn't mean you can't make it interesting for
Yes.
the animal. And don't come to me with this nonsense. But they live in burrows in the wild. Burrows are really interesting things as well. They're not
They
just
are.
a box with a hole, right?
They're
Right.
crazy
So you
interesting.
can make
They are.
you can put a little bit of effort
If
into
you really
your
want to know
cages
how
and your
burrows can
enclosures.
be interesting, that monitor burrow in Australia that they talked about,
Mhm.
13,
Oh, yeah.
13 feet
Yep.
in a corkscrew spiral with a whole bunch of false entrances and exits. Yep, it's a burrow, but
Well, it's like, have you ever seen when they pour
the
molten metal down the ant
The
hill, and you
ants.
see
Yeah, really cool. And termite mines, yeah. So don't come to me with the whole they live in burrows because it's not true to start off with, and also you can make those really interesting in itself. So,
all right,
but people
Travis,
want to
as-
know that, and they're finding that interesting.
Mhm.
Well, that's one thing I do. I like that there are so many. I like that there are a lot of different podcasts. There are several of them that I will never listen to, but I like that there are so many because there's at least one. There's something out there for everybody. Look, this is the most scientific our podcast has ever been, and will probably ever be. It is very much. We talk about food a lot, not snakes, but but that's one thing. Like you said, people come up to you. It's still weird to me when someone comes up to me and tells me they like to listen to my podcast. I'm like, why? Go listen to something else. Don't listen to me. But I do like that there's a lot of information out there, and I try to get some information out there. But Travis, as our ball python person here,
less than half of my snakes are ball pythons.
you still
I
got,
got 75 ball pythons, right? So,
No, I have 18 ball pythons out of 60 animals.
That's a lot of ball pythons, man. That's about 18 more than you probably need.
Yeah.
I mean, I say that having like 6 in the other room, which is 6 more than I definitely need. Travis, and you also are one that likes weird ass snakes.
Mhm.
Where do you like the hobby as it is now? Because like I said, it is getting more diverse, so it's got to be easier-ish to find some of the things that you like than it would have been maybe not like 20 years ago, because odd things were easy to find like 25 years ago, and then they stopped being easy to find, and now I think they're being easier to find now. But the direction the hobby's going now for you.
Well,
was a
I mean,
heavy
I
sigh,
would
dude.
disagree with you that it's easier to find the odd things that I like, because the odd things that I like are
Really
the
fucking odd.
seriously odd things that, like, I would have to go out into Namibia and collect them myself, because nobody's going to collect these things. And I'm not going to Namibia to collect animals. I do like seeing the diversity. Like, Warren said, when you've got just a monoculture of everything, it's problematic on so many levels. The diversity, not just in animals, but the diversity in topics on podcasts and things, you see it everywhere in the hobby. I think kind of collectively people are bored of the same thing over and over and over again. It got super saturated to basically where every reptile show was just thousands and thousands of 4 different species or 5 different species.
Yeah.
And it got real old seeing that all the time. So it's nice to see diversification coming out. My concern there at the same time is that people are diversifying to look for the next big major thing, which goes back to what Warner was saying, like, drop money from the damn equation, people. You know, I'm seeing a lot of people who suddenly are trying to sell egg eaters as the next big thing because they're not rodent eaters. And so everybody should be able to take care of these because now you don't have to worry about feeding your snake rodents. And I loved my egg eaters when I had them. They're awesome animals, but they're not for everybody, and they shouldn't be pushed out there as the next big thing. They're a thing that I'm sure a lot of people would like, but don't try to commercialize them as the next great big thing.
I kind of saw that with false water cobras, because they'll eat anything. People are like, you can get them to eat anything, and they're big and they're fun. I'm like, but that's not a snake for everybody.
And it's not, I think it's great that they have seen kind of a resurgence under Zac, but at the same time, I think it's a terrible thing. Because now you have a lot of people who jump into it. Well, I don't mean it as a slam against you, but like a lot of people get all excited about it.
I'm the asshole, man.
No, you're not an asshole. I think people, their excitement gets ahead of them. You when I offered you kukri, you offered me
false water.
And I was like, no, fuck no.
I would offer Warren one. I would offer Ben one. You guys want one?
We all get it free tonight.
We're good.
Yeah, you do.
We're good
We're in the
here.
mail.
I don't want to
Like we're on
do
Oprah.
with hyperactive colubrid. I just don't.
So I think the problem though is
too many people
Zac's
jump
excitement. Zac's excitement about false water cobras comes out when he talks about it.
Oh,
And so when someone
absolutely.
hears somebody talk about their excitement of whatever species, they're like, I've got to have that species.
And I think that's a
There's a
great
part of
thing.
this,
I
though.
just think the problem is that's where they stop, is exciting. And, Zac getting people to do research on things is great. I obviously advocate that because that's exactly what I made my kid do with the scrub python.
Says the person that made their kid research scrub python.
Right.
Yeah.
And like I do that with everything that I get into. I go down a rabbit hole. I have folders of just papers that I have printed out
100%
as reference material, and I go through it and I read all of it, and I really get to understand what it is that I'm going to be keeping and why I'm going to be keeping it. And sometimes I'll go deep on a species, and then I'll be like, you know what, these are really cool. I can't keep these because it's not something that I can accomplish. It's not something that I can do right. I don't think enough people do that. They hit on that excitement, and then it becomes a problem.
Oh yeah,
what
I
I
agree
do-
with that.
But there's something I do, which is when I get like, water cobra, I want your water. I send people that article I wrote for Reptiles Magazine. And then I will oftentimes send them the animals article that I wrote on natural history, blah, blah, blah. I have absolutely, I creep on everybody that wants one. And I have flat out said, you're not getting one. A lot of people,
So do people need the
get,
do we need
yeah,
the
so
do we need the flower baby version of the false water cobra? What would it be? What is the flower
Yes.
baby that
I have
you're meant to raise
no
for
idea.
a week or two before you can actually have
Oh,
one?
when you carry around
Yes.
in high school, and you have to carry around a bag of
green
flour
bag.
or an egg. Yeah.
yeah, a boom slang. That way, if you get bit, you'll bleed out of your eyes and your asshole. So you probably shouldn't do
So it's entertaining for everyone else.
yeah. But I've probably denied that easily 50, 60 people. I've sold a lot of these things to people that have ball pythons, actually. The monoculture thing, and they just want a big pet crazy snake. And it's been fun. A lot of people, actually, what's been interesting is when I do the kings and other animals, I want a pair. I don't get that. I definitely get people asking for pairs. I don't want, but I get a lot of I just want a male, because you said the males were smaller. Like I get a lot of that, which at least there's a level of responsibility there. I don't know how thick it is, but we're at least considering, so like if that person I'm going to keep a conversation with, but yeah, no, there's a little bit of,
but I think that passion though has helped the hobby too. We talked about things helping the hobby. Your passion for false water cobras bleeds out into people wanting false water cobras. We talked about Eric earlier. Look, Morelia Python Radio is definitely the reason more carpets are sold and kept in the hobby.
He's the reason I got IJs, 100%.
And look, Warren keeps all those weird ass little Central American boas and stuff like that, and those have started to take off. I've seen more people wanting dwarf boa species versus everybody. When I remember growing up, they wanted giant red tails, and now the people are wanting some of these island species and other things. So that's one thing I like about, especially with podcasting it out there, people seeing that these other things exist and getting into them, because they're really cool. A lot of these animals are really cool. You just don't see enough of them out there to see how cool they are.
Speaking of podcasts, my favorite podcast is actually called Frog of the Week, and it is about 2 minutes long every week.
Thanks for that.
I know. I'm just saying, like, there are podcasts out there for everybody. It doesn't have to be long and hard to understand.
You say this is hard to understand?
Sometimes.
Well, we'll talk about food again at some other point. All right, let's wrap this up. I need to do my giveaway real quick, and then we'll wrap it right. So our giveaway this week, for anybody still listening, is we're giving away a probe set. Again, if you've never probed an animal and you win this, please, dear God, don't test it out on your animals.
Oh no, no.
We'll talk to whoever wins this, but that is one thing. Yeah, it's one of those things that people see on a YouTube video and they're like, I can do that shit. I'm like,
My favorite is I went to a vet once and they probed it the other direction.
Wrong direction. I've heard about that. Oh,
and then I asked him, like,
Definitely
how many males do you get? And he's like, an unusual number of males. It's like, that's because you're shoving that probe up into the damn snake's rectum. Like, don't do that.
I've seen a bunch of people screw up snakes with that. Yeah, I've seen them force a little bit too hard, and they're females.
My only job is to hold the snake, and I'm nervous every time. And all I have to do is hold the snake.
I'm going to say the safest way to sex snakes is Ben.
Yeah, there you go.
You take that shit in an envelope and you send it to Ben.
Yeah.
You can't accidentally sterilize your snake that way.
This is why I breed things that I can just palpate with my thumb and go, yep, that's a boy, that's a girl.
Yeah.
That's why I like sand boas and boas. It's so much easier with boas.
100% of the time, it's correct.
Yeah, I've gotten boas in the mail before, and I'm like, that's one I have was supposed to be a female. I got it in like the day I got it. I felt it. I was like, well, that popped. Fuck. So
Hopefully it was a tree boa, but definitely one of the chunkier boas. It's fine. Yeah.
So the person that gets this, don't fuck up your snake, and if you fuck up your snake, it's not our fault. The winner for this one is James Barry. I'll reach out to James Barry. You won. Thanks to our friends over at Colossal Constrictors. You won a probe set. Please don't use it if you don't know how. If you find a friend, someone can teach you how to use it. But yes, so that was our giveaway.
Is it
for
one
this
of those
week.
probe sets that comes with like the really, really large probe as well, that you could only ever use on like a Titano boa or something like that?
Yeah.
Right?
Yeah.
Dude, it's like the thickness of your little finger.
Yeah.
I've used some of those larger probes just for other things, like for stirring things or mixing things up sometimes.
In your ear.
I've looked at that and gone, what the hell are you probing at that point?
Whoa.
Yeah.
And then some of them get so small. So speaking of real quick, speaking about sexing and having Ben help you out, someone asked something about the green tree python thing and the issue with that. I know I've talked to you before about it on here. Where are you with that one? Still trying to figure that one out?
So for a while, I did do a complex test where I was looking at some markers on the sex chromosome itself. So I would need to have if you're like trying to sex a clutch, I would have to have the sire and the paternal grandsire or a known male offspring from the same sire. And then I could do the sequencing and just see whether it got the same male chromosome or if it got the other one, then I knew it was a female. But to get one that works across 50 or 60 different known green tree python samples, I haven't gotten one to work across many where it's like a single shed test.
Yeah.
I know that's something that Warren has spent some time on too. I don't know how well that test works for you.
The sex chromosomes of Morelia turn out to be really complex. And that's all I can say right now. So the likelihood of us having a shed test that's going to be working 100% of the time is we're still a little bit further out from that there.
Yeah, I think
The
everybody
sex
looks at.
chromosomes are just odd. I will say that there, and the ones that when we find a marker and we're convinced it's male, and then we sex a whole bunch of them to confirm, and then you find females coming up as male.
Are they
So
ZW?
the sex chromosomes are really more complex than, and we're working on that in a collaboration. So not to develop a test, but to understand the evolution of sex chromosomes. But they're really funky.
Green
what
trees,
I'm
are they
hearing is there's going to be a real cool paper at some point.
So green trees, are they the ZW? They're not XY, right? They're ZW, and
They're XY.
They're XY. Okay, I didn't know if they were.
All boas and pythons are XY except for the Madagascan, and also we're not sure about the Candoia. They might be ZW as well, but the Madagascans are all ZW. The boas and pythons evolved XY sex chromosomes independently. What was that, Travis?
I've got 5 Candoia I can get you sheds for.
Yeah, we need more than that.
Well, that's all I can give you right now.
Yeah. See, you're not helpful. So thanks a lot, Travis. So. And also, we're not really doing any of that work anymore. We're doing a chromosome level boa constrictor genome, but that's for another project. But we're not really doing parthenogen testing and all that kind of stuff. That's all. Partially.
Did you have to bring your
thing of the past?
Did you have to bring your cooler of frozen animals with you all the way to Virginia Tech?
Yeah, they're in a -80 freezer that I've got in the department. Yeah, or in my lab. And then I'll find a way of disposing a lot of that stuff. Yeah, yeah. Because we're just not, with my new position, it's not as broad as what my last position was. So I don't have the flexibility to work on a lot of the parthenogenesis stuff that I was working on, and I don't necessarily want to anymore. I think so. I had to turn down a couple of people this week about testing their parthenogens, because they want a paper from it. And for me, a paper describing parthenogenesis in a species of boa or python that it hasn't been found in before is just stamp collecting, and I'm not really into that. I'm into it if it's a new lineage that it hasn't been found in, or really understanding the mechanisms, and we've got papers coming out on those. But not just I'm past the whole, here's another, here's parthenogenesis in a green tree python, or here's parthenogenesis in a Amazon tree boa. That's just, it's so widespread throughout the snake phylogeny that it's no longer exciting.
Just,
to me, at least.
Make you a little extra money. Any of those animals you have to get rid of, just sell it to those people that sell the dead animals in jars at reptile shows. Just start selling those.
I've actually contacted some of those people. Yeah, they've contacted me because I've got a lot of snakes in my freezer. The only thing that I want to keep is I've got half of a Gila monster, and I want to keep that in my freezer because I need to find someone with the beetle colony that can sort that out, because I need
Travis.
Travis,
there's your chance to meet Travis.
You do.
Travis,
I've got a beetle colony.
There it is.
I'll have to. I will
bringing people together.
I'll decapitate it, and I'll work a way of getting that to you, because I want the Gila monster skull is really cool looking.
What he didn't tell you, Travis, is it's the back half. It's just the asshole. You need to clean out the asshole.
Yeah. I just want the anus. No, it's the front half. So, and that's right. Alicia's got one as well. I'll be in Knoxville. I'm just looking at the chat.
Hey, he said, "Fuck you, Travis." Someone else has one. He says he knows you're closer,
and I'm
but never
more
mind.
likely to be in Knoxville in the next couple of weeks than I am in Travis's. Yeah, I'll send him the back half, and I'll send the front half to Alicia. I'll compare quality at the end.
If you want people to get a hold of you,
well, Warren doesn't. Zach, if you want people to get a hold of you,
yeah.
What is the best way? And go ahead and plug your school and make sure everybody realizes it's not Liberty University. That's a different.
It's not Liberty.
Oh God,
it is West Liberty.
Yeah. So, how can I get a hold of you? What's your podcast and everything?
That's weird. To get a hold of me, Facebook and Instagram. Zac Loughman on Facebook. That's the best one. And then podcast is Colubrid and Colubroid Radio. On the same network as Warren. So Morelia Python Radio Network.
Just need Warren to put out more episodes.
Mm-hmm.
We're starting. It's my fault.
But
he's
you've
a little
been
busy.
moving. I think you've got.
I was going to say, didn't you just move?
Don't give him excuses.
No, he deserves it.
I like boas, and I
moving
want to hear
a
about
whole
boas.
lab is not a trivial
An easy
thing.
feet.
He just
We have
moved
3
a
episodes
freezer
lined
full
up.
of dead stuff. Warren, just go ahead and just plug your podcast. They don't have to get a hold of you. You don't have to talk to people.
No, they can't. It's I don't mind people emailing me or contacting me. So the podcast is Boas on the Morrill Python Network. We're recording our next episode January 4th, and then we've got a bunch lined up after that. As I said, it was my fault that we weren't recording because I was just everything with the move. To get hold of me, also you can get it through my Instagram, which is Boa Booth, and through Facebook it is Warren Booth. Don't send me a friend request on Facebook if I don't know you, because I'm not going to friend you, but I will happily answer your questions in most cases. Other than that, I think that's it.
I love that you have the best business name for someone to do reptile shows who doesn't do reptile shows.
I know, right? I've thought about it. I think I will be soon, just because of the diversity of animals that we now have. From the Sanzinia to the emeralds to the Amazons to all the different boas, and then I just got into rainbow boas in a big way, and then we've got a bunch of Antaresia morphs and all that kind of stuff. So I think, and then I've got a bunch of carpets and a bunch of womas and some colubrids now.
Just starting to
I think
remember
in time
things.
I will. The difference is that whenever I go to reptile shows, which is very rare, I like to go and have a beer and walk around and talk to friends.
Yeah.
The idea of standing behind a booth for, whatever number of hours per day sounds miserable to me. So
You need
but
somebody.
I think I will.
You
Just
need
feel like
somebody to come run your table for you.
No, I just feel like it would be Warren telling people, no, you can't have this. Keep going.
No, I'll happily sell it to you. I'll
Keep
sell
walking.
you anything you want. Just show me the money. I'm in it for the money. I'm not in it for the snake.
That's what everybody's learned from this show is that Warren is
I've
just
been doing it
in
for
the
30 years, just for the money for 30 years. Yeah. It's worked really
Just-
well.
Oh my god.
Boa Booth is just a great name for a. And he's never had one. Travis,
If anybody
we'll
wants to
come
make me
back.
a free logo, go ahead. Yeah.
Jeff, if you're out there, you can hear that. Who's made all our logo? Feel free to do it.
I said free.
Free. We'll skip Travis. Come back to him. Ben, go ahead. Tell people how they can get a hold of you and send you sheds full of shit and urine and nasty stuff and things that you're gonna do miracles.
Keep it clean, people. Keep it clean. We don't need that shit. So Rare Genetics Inc. We're pretty easy to find on Facebook or Instagram. We also have a weekly show, Reptile Genetics Weekly, that usually comes out on Tuesday nights. But since I'm setting up a big run for shed testing today, and I was coming on this show, we'll have our episode this week coming out tomorrow. But we're easy to find on YouTube too.
I appreciate your show because it allowed me to pull a lot of pictures of Travis's face for all the stuff that I made this week. It was great.
Are any of these podcasts kid friendly?
Ours is.
Yes.
There's
Ours.
Yeah.
It's just our
you.
podcast.
It's just you.
It's me.
Yeah.
I have to be kid friendly for a majority of the week. This is
Yeah,
not
but
what I
my
choose to be.
zoology club kids want to listen to podcasts.
Then I
and
guess it
can't
sounds like some-
recommend ours.
I guess it sounds like somebody needs to make their own damn podcast.
I don't have time for that.
Exactly. Travis, go ahead and tell people how you don't ride motorcycles for a living.
Yes, I don't ride motorcycles for a living. Don't go on Facebook and find the motocross racer. Should be pretty obvious it's me. I've got a snake in my profile picture. Also,
Is it the black
Instagram.
shirt picture?
Yes.
the black shirt
I
picture.
love that picture.
Credit to my wife. She's the one who set that one up, and she's the photographer. So
She did a great job.
yes, she did.
Yes. The green tree looks great. The guy in the picture, not so much.
You can also find me on Instagram, Snakes and Bakes.
He does a lot of weed. Smokes a lot of weed.
Whatever.
Yeah,
Dude is always high.
That's awesome. Think of that. That's a great business thing.
Move to Colorado and open
Not
up
what I was
a.
expecting, Travis. Not what I was expecting.
No.
wrong bakes.
It's weird. Travis will get drug tested at work tomorrow. It's
Oh
all.
my god.
I probably will be, but that's okay because I always pass.
It's part of the
Yeah,
court
you need-
ordered mandate. Don't worry. It's fine.
That's just conditions of my job.
If anybody wants to reach out to us, we are the Reptile Gumbo Podcast on Instagram, Facebook, at gmail.com. Don't forget, save 15% on VivTech. Use code Gumbo22. We will be back in 2 weeks. We're taking next week's Christmas. We're not going to be on here for Christmas. So we'll be back in 2 weeks. I actually do have a guest lined up for that one, so we can breathe a little bit. Those of you that have podcasts that have guests, have you found that to be the hardest thing to do? Is find people that want to come on and talk?
No.
My problem. It's not a huge problem. It's not a huge problem
just
here.
You, James.
No, it's at shows. It's at shows. I find reptile people are like, because reptile people are not people in general.
Yeah.
And they're like, I'm afraid to talk. I'm like, it's just talking. Like I'm not asking you to do anything. There's not going to be people there like laughing.
No tricks.
It's just talking. And then once they're on there, like, oh, this wasn't that bad. I was like, it was literally talking. I don't know why you thought it was bad. It's.
I've had 2 people.
I think the key is.
They know. Everybody else has been yes.
I think for me it's if you just do it really infrequently, then it's a lot easier to find guests. You're asking fewer people.
When you're almost at 200, James.
That is true. I've almost said 200. That is way too many people. That's why I've had Travis on like 70 times. And then people tell me to quit having Travis. Oh, it's just me that says to quit having Travis on.
Whatever.
You just don't like that I make you read, James.
Reading
he's
is
your
not
favorite.
fun, and if you could turn everything into a small YouTube video,
It
then
won't
I'm
hurt
good.
you to read.
Or a meme. That'd be even better.
A meme's
Yeah,
even better. Just everything can just be
I'm
a
gonna
meme.
stop letting you buy books when we go places.
I've got a
'cause you
whole
don't
bookshelf.
read.
Look at all these books I bought.
I know.
This one's on Gila
You're not
monsters.
actually gonna read them. Why am I spending
I'm not
money
gonna
on
read
stuff
it.
you're
This
not
one's
gonna
on
read?
Poisonous Snakes of Texas.
Which Gila monster one did you get?
Yeah, what Gila
Hold it up.
book?
Facts and Folklore of America's Aztec Lizard by
I
David
will
E.
read
Brown
these
and
before
Neil
he
B.
will.
Carmody.
Huh.
It's from like I tend to go to
All of his stuff
half-price
is
books.
at least 1950 or older.
No, this one's
Got to
not.
be an older one.
It's an older one. It was 1991 was the last time this was.
Okay, well maybe not 1950.
They describe that as
That was a
older.
bit dramatic.
Yeah.
Bit dramatic.
I just like going to half price books and buying like the old books of old reptile books.
I'm starting to get antsy. The steroids are kicking in again.
That's right, Katie. The steroids. I forgot to. Sorry, she's training for the Olympics.
Yeah. Yeah.
So if anybody wants to reach out to us, we'll see y'all in 2 weeks. Thank y'all for coming on. Wait around for a second. Goodbye, everybody else that was watching. Good night.